Institute is mandated to coordinate and supervise research, development and manufacture vaccines in Ghana.
As part of our mandate is also to facilitate the research and development of academia and industry.
As Ghana forges ahead in her quest to be a known vaccine production hub, it comes along with hope, self-reliance and better healthcare for the entire continent.
It feels so good to be a Ghanaian at this time, knowing very well that Ghana has taken up this bold initiative to produce or manufacture her own vaccines for the well-being of her people and also for the rest of the continent. Reporting for GBC News, Gifty Ajay, Pram Pram, Greater Accra region.
All right.
In the heart of
Good
Ningo
afternoon,
Pram
everyone.
Pram in the Greater Accra.
Grace, give me a thumbs up if we're ready to go. 5 minutes. Okay, thanks. All right. Good afternoon, everyone. We're quite behind schedule at this point, 35 minutes. But I think we can make it up very easily if everybody sticks to the time that they're allocated and the speakers are here when they're supposed to be. This is my favorite part of the conference because this is the beef of the conference. This is why we're here. And to kick it off, I'm going to introduce the chair of our graduate committee and the incoming HOD of the Department of Biochemistry, Cell and Molecular Biology, Professor Niels Kwashi and Professor Teresa Manfojira, to chair the first session.
Maybe I would have to use my natural voice, which I think is strong and deep enough. Peter Kwashi introduced both of us, but for us to be very sure who is who, I'll ask my colleague to introduce herself. After that, I will also introduce myself, and then business starts.
Good afternoon, everyone. My name is Teresa Manfojira. I'm sharing with
Professor Niels Kwashi. So this is the real business. The business starts now. So without much ado, I would want to invite our first speaker. The preliminary section takes 50 minutes plus 5 minutes questions time. And the fellow section takes 12 minutes, 3 minutes questions. And so without much ado, I invite our first speaker for the preliminary section, Kamara Abdul Salam. Okay, so please go ahead straight because we are behind time. Straight to the. You have 50 minutes and 5 minutes questions time.
Thank you. Can you please help for the slides?
Slides, please. The slides.
And what about mine?
Okay, so okay, thank you. Okay, so good afternoon, everyone. First, I would like to thank the WACCBIP for giving me this opportunity. I think it's working, right? Okay. Okay, so it's really great to be here. So this is my second, I think my second research conferences in Africa, but my first one in West Africa. And I'm very happy to be part of the WACCBIP now. But today I'm going to present to you my postdoc work over the past 3 years in Denise Calado's lab at the Francis Crick Institute in the UK. So you may know that the bone marrow is our blood cells factory inside our bone, so that produce various number of cells every day. So the bone marrow produces roughly up to 600 billion blood cells daily, depending on the body's needs. So it is a very complex environment. Besides this complexity, you will see that some cells can manage to find a sanctuary to survive in the bone marrow. So bone marrow is the site of the hematopoiesis, and B lymphopoiesis is part of the hematopoiesis, which is the process where mature and naive B cells are formed in the bone marrow. So when B cells are formed in the bone marrow, they migrate to the secondary lymphoid organs, such as the spleen here. So when they encounter the antigen or pathogen, so they will get activated and differentiate to the plasma cells and memory B cells. And the plasma cells migrate to the bone marrow, where they can survive for days to decades, depending on the circumstances. So plasma cells are the key to the success of vaccination and protection from infection. You know, plasma cells migrate to the bone marrow to survive, and many cellular components and cytokines have been reported to be very critical for plasma cells' survival in the bone marrow. But however, we still don't understand well how plasma cells exploit the bone marrow to survive for decades. And this spatiotemporal organization of plasma cells in the bone marrow is not well understood. So I want to emphasize on the importance to understand how plasma cells are organized in the bone marrow, because this is critical to understand not only how long-term protection is maintained, but how these cells lose control in malignancy, such as multiple myeloma. So I'm not going to talk about multiple myeloma today. So in 2020, in Denise lab, so we have generated a mouse model, which I will call JCHEN here for simplicity. So in this mouse, we have inserted a tamoxifen-inducible GFP tagged cryocombinase in the endogenous locus of JCHEN. So JCHEN is highly and specifically expressed in bone marrow microenvironment. So when I joined Denise labs, I developed a high-resolution 3D imaging of the bone marrow. So we can see a simplified protocol here. So we immunize animals and we label the plasma cells. So then we trace plasma cells over time. So early time point, 5 days after we label them, 2 months and 5 months after. So you can see here in this movie, so red cells you can see here are the plasma cells in the bone marrow. And the green is the vasculature. So you can see there is a lot. And they also stand for the pan-Ig, pan-immunoglobulin markers. So this was very interesting. So now we have used some image processing tools and algorithm now to characterize the crosstalk between plasma cells and the microenvironment. So in this movie, you can see that there is an heterogeneity in plasma cell localization relative to the blood vessel. You can see some plasma cells closely touching the vessels and other plasma cells living farther away. And in this figure here, 5 days after we labeled the cells, you can see that heterogeneity with some cells in blue here, the one that are touching the vessels, and the green one being farther away from the vessels. And this heterogeneity is preserved over time, as you can see here, with a significant increase of the sorted distance of the cells relative to the blood vessels as they become long-lived. So this is color coding of plasma cell sorted distance to the blood vessels. So now we have used some mathematical model to understand better the interaction in plasma cells. So we calculate here the probability density distribution of plasma cells relative to the blood vessels. So interestingly, you can see early after we labeled plasma cells, you have every red line here is the distribution of plasma cells from one mouse. So you can see early time point, you have two populations, the population of the cells near the vessels and the one living farther away. But as they become long-lived, so you lose the population near the vessels and the cell will be accumulating away. So the vertical line here is the attraction distance, so which tells you that the distance from which plasma cells accumulate away from the surface of the vessel. So this is very important to understand this attraction distance for the rest of the talk. So 12 micron attraction distance plasma cells, which means that before 12 micron, plasma cells are repelled. After 12 micron, plasma cells are attracted relative to the blood vessels. So, and we quantify the proportion of the cells before and after the attraction distance, you can see over time. As the plasma cells age, the majority of the plasma cells will accumulate after the 12 micron, after the attraction distance. So which tells you that long-lived plasma cells accumulate away from the perivascular environment. So, you know, there are different types of plasma cells. You have IgG, IgA, and IgM plasma cells. And IgA plasma cells have been reported to live longer than other plasma cells. So we wanted to understand whether the isotype of plasma cells can be linked with their lifespan. So we scan the bone for the different isotypes. So you can see here, the blue are the IgM plasma cells. So their relationship to the blood vessels, so they are touching the blood vessels. If you look at the red one, they are living farther away. So, and this is the quantification or the probability density distribution with the peak of IgM plasma cells near the vessels, while IgA plasma cell will be peaking farther away. So this plot is somehow showing what is already here. But interestingly, if you look at, we quantify the proportion of each isotype before and after the attraction distance, you can see that before the attraction distance, so in the perivascular, the majority of plasma cells are of IgM isotype, while after the attraction distance, the majority of them are for IgA isotype. And interestingly, you can see that nearly 80% of IgA plasma cells are located after the attraction distance. So that was really very interesting. So basically, you have two niches in the bone marrow. So you have the perivascular niche, which you can see near the vessel, the IgM plasma cells closely interacting with the vessels. And we have also the deeper niches. What I'm calling the deeper niches here is the cells living farther away, as you can see here for IgA plasma cells and also somehow for IgG plasma cells. So there are two niches. Now the question is, are there some biological biomolecule underlying this differential localization of plasma cell isotype? Is there any implication for cytokines or adhesion molecules? So to understand this question, we performed a digital spatial profiling of the perivascular niche and the deeper niche. So this is spatial transcriptomic of the tissue section of bone marrow 5 months after we labeled the plasma cell. So we are looking at very whole plasma cell here. You can see the red. So we select long-lived plasma cell area, and we also select the perivascular area. So then we get the transcriptome of both areas, so then we can compare. So you can see we have done differential gene expression. And interestingly, if you look at in the deeper niche, we found some genes that are upregulated, so which are in red here. And the blue one are the ones that are upregulated. So interestingly, we could identify some genes, which, for example, CD37 is already been published to be very critical for the maintenance of long-lived plasma cells. And also CXCR currently have lower expression of CXCR4 at protein level, and this is also confirmed at gene expression level from the single cell data of bone marrow plasma cells. And then we test this functionally by in vivo inhibition of CXCR4 with a drug which is known and highly used in, for example, in cancer treatment. So we inhibit CXCR4 by treating animals accordingly and analyze the proportion of the different So now we are generating genetic models where we specifically delete CXCR4, specifically in plasma cells, so using the J-chain background, or overexpressing CXCR4 in plasma cells, and then we now look at the impact on the different isotype in terms of cell viability, retention, and antibody production. So, in conclusion, using pioneering in vivo mouse model, high resolution 3D imaging model, and spatial transdermic. So I hope I could convince you that we are shedding light for the first time in bone marrow plasma cell positioning and demonstrated for the first time that plasma cells positioning in the bone marrow is not random. So long-lived plasma cells accumulate within the deeper niche, meaning away from the hematopoietic niche, at a distance of 12 micron from the perivascular niche, and we have a maximum accumulation at 7 micron away from the vessels. IgA plasma cells accumulate within the deeper niche, which is the long-lived plasma cell niche. And the spatial transcriptomic former, I'm still part of the lab. So in the UK, Denise Calado's lab, which I would like to thank everyone, and especially Denise. So Denise is here, is giving another talk, but it will be on big lymphoma, so please enjoy it. And Tony is a PhD student who is taking over this work, this interesting work. Because I've moved to a new position now at the WACCBIP as the African Career Acceleration Fellow of the Quick Africa Network. So this is my new home, my new place. Is it beautiful, isn't it? So with the mentorship from Professor Awandari and Dr. Denise Calado, so in the next 4 years, so I'm gonna be investigating the plasma cells biomarkers across different vaccination here in Africa, in Ghana, which we can use to predict the vaccine efficacy. So with that, I would like to thank everyone and thank you for your attention. Happy to answer your questions.
Dr. Kamara, thank you. Thank you for your stimulating presentation, for sticking to time. So, because of time, we'll stick to 2 questions. So if anyone has a question.
Yes, Gina has a question.
Can you have the...
Okay.
I can hear you.
Try to.
New plasma cells coming into the bone marrow affect those niches. So you imagine in a pathogen case where you have lots and lots of new, probably long-lived plasma cells going to be made, going into the bone marrow. And we know that long-lived plasma cells aren't eternal plasma cells and eventually will get kicked out. Do you know what the dynamic is with new incoming cells and getting rid of the different niches?
That's the interesting question. You have noticed the J-chain model label all the plasma cells. So to answer your question, so what I propose to do is we are generating a mouse model for like malaria parasite. So or other antigens, we can trace antigen-specific plasma cells because we have the capacity to label the cells. And if you couple this with the antigen specific, so then we can look at first vaccination or infection, where are these antigen-specific plasma cells? And if you give them again the shot, how it will disrupt the niche. Yeah.
Any other question? Okay, thank you very much. Let's give thank you.
So we now go on to the fellow session, and we will start with Franklin Nwakam. And he will talk on targets of CSP-based malaria vaccines, what we missed in 1987 and what is missing now. Please, you have 12 minutes.
Have you started my time? Good afternoon. My name is Franklin Nwakam, and I'm here to give a presentation on work that we've been doing in the lab, and it's titled Targets of CSP-Based Malaria Vaccines. And I'll be talking about what we missed in 1987 and what we are missing now. Malaria is a disease of major health concern, and every year several people die and a lot more fall sick. And the at-risk group are children under the age of 5 years and pregnant women. There are 5 species that cause human malaria. We have Plasmodium falciparum, which is the most deadliest, Plasmodium malariae, and Plasmodium ovale, which were thought to be benign parasites. But work in our lab has shown that even in clinical samples, we are having these parasites pop up. And these parasites have started causing severe disease and even death. We have Plasmodium knowlesi, 40% against severe disease, and R21, which has an efficacy of about 70%. And these are all big. It has a molecular weight of about 58 kilodaltons, and it covers the entire surface of the parasite. It is the most dominant protein, and it consists of, and it has a C terminus devoid of the GPI anchor, conjugated to hepatitis B surface antigen. And this is expressed in a system that already expresses free hepatitis B surface antigen. So in a vaccine formulation, you have these two constructs in there. The R21 just removes the free hepatitis B surface antigen. And the reasoning behind this design is that studies have shown that immune responses against the CSP are mainly against the repeat region. However, these were very few studies and they were limited tools. So we decided to go back to ground zero, and for this time we are considering the 2 other Plasmodium parasites that are especially circulating in Ghana, malaria and ovale. And we are looking at CSP recognition by antibodies from naturally malaria, which is recognized even by single steady samples. And after detecting these antibodies, we have a high falciparum transmission. And as studies like in Tampo, actually we typed these population, we had 6.7% falciparum and 0.7% ovale. But with the antibody levels, we have similar anti-PF-CSP IgG titers compared to anti-PO-CSP IgG titers. And together with work by Guy et al., McMahon, and Chatterjee, we are inclined to conclude that this dampened anti-CSP, PF-CSP response is mainly due to antibody feedback inhibition underpinned by epitope maxing. And these are usually enhanced by tandem immunogenic repeats, which are characteristics of the CSPs, protein conservation, hyperexposure, and intrinsic disorder. Yeah. And then we made peptides. We synthesized peptides from the CSP domains, 20 amino acid long peptides with 10 amino acid overlaps, and we used dot plots to identify, to detect recognition of antibodies against these peptides. With PF-CSP, surprisingly, we saw recognition at the N terminus, which is thought not to be immunogenic, and the C terminus together with the repeat region. And we saw this for PM-CSP and PO-CSP also. So we tried to establish a hospital and community cohorts. And the information this can give us is that if you have a population such as Medina is just around, and you assume parasite exposure at almost equal levels, which is quite reasonable. And at the end of the day, you have some of them falling sick, malaria, going to the hospital, and some not falling sick in the community. We classify those who fall sick as the hospital samples, and we are inclined to name them at risk cohort. And we have the community samples, we're also inclined to name them a protected cohort. We quantified anti-CSP specific IgG titer against these peptides using ELISAs. What we realized for PFCSP is a high and diverse antibody recognition across peptides from all the domains. We have peptides from the N-terminus, the repeat regions, and the C-terminus. What we see is equal antibody levels for all these. But for the minor repeat, we hypothesize that antibodies that bind to it affinity mature to bind to the major repeat. That's why you have limited amounts of those. But in the hospital cohort, you see a polarization of IgG response to the major repeat and the C-terminus. We see this for PMCSP also in both the community and the hospital. There's a polarization here. And for POCSP, we see a similar polarization in the hospital cohort, whilst we see recognition in the N-terminus in the community cohort. What our results show is that we have recognition of all CSP domain by naturally acquired IgG. We have a higher and more diverse antibody response in the community compared to hospital individuals. And hospital individuals have a polarized immune response to the C-terminus, to the repeat region and the C-terminus. And these suggest that a polarized anti-CSP immune response may be a feature of an immature immune response. And the anti-CSP N-terminus immune response may be important in protection. This is critical because the RTSS and R21 vaccines do not have the N-terminus. Cross-species protection is desirable, so we have a vaccine formulation that protects against multiple species. And with a paradigm that's proteins that perform similar functions will have similar structures, and this is underpinned by similar protein sequences, which can be platforms for the development of cross-reactive vaccines. We sought to compare the sequences of our steady proteins. At a full protein level, we realized very limited protein sequence similarity, which is surprising for an essential protein like the CSP. But however, we hypothesized that sequence conservation may be domain-based. So we did that for the N-terminus and the repeat region, which recorded similar amino acid sequence similarities like the full-length protein. But with the C-terminus, we see a higher sequence similarity. By just scanning the sequences, you see the conservation of the pixel motifs that are repeated twice, the protease cleavage site and region 2+, and these have been shown to be essential. With a structure, comparing the full-length structures, you don't see significant sequence similarities. This is the TM score is between 0 and 1. 1 means the structures are perfectly the same, and 0 means they are not similar. So usually we use a cutoff of 0.3 before. An essential protein like the CSP, this is quite low. And so with our hypothesis, number one, we missed the addition.
Thank you for a really excellent talk. I disclaim I'm not a malaria expert, but do you have any idea about longitudinal development of these different antibody responses? So for example, in your community group, you saw N-terminal responses were dominant, and your hospital group, you saw C-terminal responses were dominant, and the repeats were mixed between the two. But do you think that there's a contribution of the hospitalized people being infected for longer, and so if you looked earlier, you would also see those N-terminal responses in them?
Yeah, that's a very good question. So first, you asked about the longitudinal exposure. So for our cohorts, the community cohort is mainly made up of adults, and it's reasonable to hypothesize that these people have been exposed much more longer. Yeah. And then your second question is about that's okay, right, yeah. So that's you okay? Yeah, we have hospital samples that we took longitudinally, but they are not best for answering this question. But I think we can have a look at that later. Thank you.
Okay, thank you. Additional questions? Okay, please go ahead.
Yeah, you go ahead.
Yeah. Yeah.
So you might want to check, I don't know whether you check to see whether there is some balance in distribution
Yeah.
between those two, because you might want to
So that's an excellent question. We actually did that, and we saw the same stuff. Yeah, yeah. So we took the children out. There were still a lot of adults, and we compared them, and it was the same. Yeah.
Yeah, Jane.
Yeah, my question was more or less on the same. I think
Yeah.
it's the comparison of the hospital cases versus the community. They're different ages,
Yeah.
and you might argue, I would argue the other way around, that those in the hospital have a immature response because either they've not had as much malaria
Yeah.
and they've not been exposed for as long, so their immune response or their antibody response is against the immunodominant parts of the molecule, whereas in the community you have an older population who's had longer exposure and will get all the subdominant responses as well.
Yeah.
Is there any way you can compare just within the community between those that are, say, symptomatic and those that are not, or something like that?
Yeah, we can do that. But for this sample, the community cohort, they are all PCR negative. So we don't have symptomatic, asymptomatic. Yeah.
But I think it's the length of exposure they've had to the sporozoite that determines the repertoire of antibody responses they can make.
But in the hospital cohort too, we have adults, and that's a good thing about these two cohorts. But even when you take the children out and compare just adults, you see the same stuff. Yeah.
All right. I think we can move on. Thank you very much, Franklin.
You're welcome.
So our next presenter is Angela Ayiku, and she'll present on overexpression of heat shock protein is inversely related to viral yield and viral apoptosis gene expression. So the slides are delaying a little bit, but I'm sure it will be up very soon.
Hello, everyone.
Yes.
My name is Angela, and today I'm presenting on the topic overexpression of heat shock proteins 90 and 47 is inversely proportional to viral yield and viral apoptotic gene ORF005L expression. So a few years back, there were reports about a high mortality outbreak, viral disease outbreak in cage culture tilapia farms on the Volta Lake. And this led to the fold-up of so many farms and also devastating economic loss. And in fact, Ghana lost about 130 million USD during that period because of this outbreak. Now, the virus of interest is a large icosahedral virus from a megalocystic virus from from the family Iridoviridae. And normally these viruses are double-stranded DNA viruses. And even though the virus has a broad host range and a broad geographical distribution, this was the very first time it was being reported in Africa. So from our previous studies, we realized that the entry of this virus was probably from a single source through illegal importation of fish species. And this is because the sequences we had from the sample spanning across the lake had 100% homology with the MCP genomic region, and they all had a common SNP. but recently we found new SNPs which indicates a gradual divergence from the ancestral strain. Now, during this outbreak, a lot of farmers were using some interventions, and the ones that stood out most was a commercial vaccine rolled out by the Ghanaian government and a technique known as heat shock treatment. Now, these two are very contradictory in the sense that vaccination has to do with the host being introduced to the viral antigen prior to a primary infection, whereas with the heat shock treatment, what the farmers were doing was actually deliberately infecting the fish with the wild-type virus and then exposing them to heat stress. Now, I don't know if the farmers knew the science behind this, but they attested to it that it was really improving their survival rates. And in fact, when we did our studies, we also found out that the farms that were practicing heat shock treatment were improving their production rates. And to be honest, this is not a new phenomenon at all. This is actually something that's old age. It was found that prokaryotic and eukaryotic cells actually produce a substance known as heat shock proteins when they are under environmental or physiological stress. So these heat shock proteins naturally play a role in proteostasis, cellular transduction. They inhibit the intrinsic and extrinsic apoptotic pathways. So coming back to the pathogenesis of the virus and how it may be linked to these heat shock proteins. Now, the virus has been found to induce autophagy once it infects its host, and then also induce apoptosis during some part of its infection. So the question is, why would the virus induce autophagy, which is a process that's meant to degrade the virus at the end stage? So that it means that the virus is definitely benefiting from this autophagic process. So our research question was, what role does heat shock treatment play in reducing mortalities in an ISKMV-infected tilapia population? And how does this modulate the pathogenesis of the virus? So our aim for this study was to determine interaction of heat shock proteins, viral replication, and apoptosis. So first of all, what we did was to develop a primary cell line just to be able to mimic the pathogenesis of the host. So we developed a primary cell line from the brain tissue of the Oreochromis niloticus, and then we confirmed this with Sanger sequencing to be sure it's the strain of interest. And then we carried out a TCID50. So when you look here, this is what the cell line looked like after 14 days of propagation. And so after developing the primary cell line, we used it in an in vitro challenge experiment. So with the challenge experiment, we had 4 different groups. One group was infected with the virus and then exposed to 38 degrees Celsius, which was 10 degrees higher than the ambient temperature for tilapia farming. And this was done 48 hours after infection. Then we had another group that was infected with the virus, but then there was no heat shock treatment. And then we had a third and a fourth group. The third group was mock infected, and the fourth group was uninfected. So from our in vitro results, we realized that at 48 hours, the response was quite immediate. Heat shock proteins, gene 90 and 47, were overexpressed after the heat shock treatment. And we realized that 72 hours after infection and 24 hours after heat shock treatment, the viral load, the viral yield was significantly higher with the untreated as compared to the treated. And then also we realized the apoptotic gene was also overexpressed with the untreated as compared to the treated. So this was really interesting. So we tried to corroborate what we were seeing. So first of all, we also took some images of the cell culture. At 24 and 48 hours, there was no remarkable CPEs in any of the groups. But then at 72 and 144 hours, we realized that the infected group had very significant CPE as compared to the challenge. So basically, for our findings for the in vitro challenge, we found out that there was a significant fold difference with heat shock proteins. There was an increase in extracellular viral yield with the untreated as compared to the treated. there was overexpression to the treated. Had a control group. Now, we also took some data as well from these control groups after 6 weeks. So for every 2 weeks, we took some parameters. So first of all, we looked at the survival rate. Now for 42 degrees Celsius heat shock group, all the fish died because the temperature was too high for their survival. But the interesting thing is, at 40 degrees Celsius, there was very high survival as compared to the control group, which is D. So over here, you realize that at T4, that's after 6 weeks, the survival rate was less than 10%. But with the heat shock group at 40 degrees, the survival rate was about 70. And the same for heat shock at 38, the survival rate was about 40%. But what was interesting was that the growth rates showed a different result. So when it comes to the growth rate, you realize that the control group had a better growth performance as compared to the 40 degrees and then the 38 degrees. But this could be explained because they had more food. I mean, there were less fish left in the tank. They had more food and all that. So we also realized that there was overexpression of heat shock proteins once again in these fish. And then the viral load was relatively the same throughout all groups. So we came up with a suggested mechanism. So when the virus infects the cell, it induces the autophagic process, utilizes the glucose metabolism, and then induces apoptosis to avoid the degradative stage. But then with heat shock proteins, the heat shock proteins are going to inhibit both the intrinsic and extrinsic pathways of the apoptosis. So basically, these viruses will be degraded in the autophagic process. So in conclusion, we would say that this study demonstrated short-term effects of heat shock proteins on ISKMV replication in the natural host cell. And this is a baseline study for further understanding of the mechanism behind heat shock treatment as a control measure. Heat shock treatment is capable of slowing down disease progression, but not completely clearing the virus from what we just saw. And the intervention may lead to persistence of the virus on the lake. So I've suggested a few studies that we can look into, like protein expression and binding activity of heat shock proteins during viral replication, long-term infection of heat shock treatment on growth rate of the host, and then also explore vaccine targets by using the antisense oligo approach. So I would like to acknowledge all these people, and I have a picture over here of University of Ghana just to re-emphasize how much work we can do when we collaborate, because this was a collaboration between the Department of Marine and Fisheries and WACCBIP. Thank you.
Thank you very much. We have time for questions. Yes.
I think I have a lot of questions for you. So my question is that are you considering looking at innate immune responses? Because we know that HSP90 has got something to do with protecting innate immune genes. So maybe it's possible that when these proteins are overexpressed, they will protect the innate immune genes, which may be counteracted by some viral proteins. I don't know much about this virus, so I don't know if you are considering looking at that.
Yes, actually HSP90 does a lot. With all the heat shock proteins, they do a lot of work within the cell. They even interact with transcript proteins. So yes, we can do more studies to understand the interactions between the HSP and the virus.
Any more questions? Okay, so I think we don't have any additional questions. Thank you very much, Angela.
So we'll next invite Frank Osei. So he's presenting on effectiveness and immune checkpoint marker kinetics of SARS-CoV-2 booster vaccines in Ghanaians.
Yeah.
Frank.
Okay. Yeah. Thank you very much. So my title, like Retty said, is to assess the effectiveness and then the longevity of the COVID-19 booster vaccines that were administered within the Ghanaian population. Yeah. So this will be the outline. Okay, coronavirus causes the COVID-19 disease. And like we are all aware that this disease causes a lot of pathology or morbidity and mortality within the global sphere. So the WHO estimates that about 6.9 million deaths were associated with this COVID-19 disease. And the viral structure is such that it has proteins that enables it to carry its activities. And one of those proteins is the spike protein that enables it to be able to attach to the human cell and then enter through the subunit 2 of the spike protein. And once it gets into the human cell, what happens is that it has the nucleic acid protein that enables it to be able to multiply and increase its quantity. In Ghana, we also had our share of the death. And that was, the Ghana Health Service estimated that over 1,462 persons died due to this COVID-19 infection. Now, while the disease was prevalent, to be able to curtail its morbidity and mortality, vaccines were very crucial in its in managing or in it preventing its spread. And the African CDC estimates that 646 million vaccines have been administered in Africa. And also in Ghana, the Ghana Health Service estimates that over 28 million people had received these vaccines to be able to mitigate the impact of the infection. Now, we know that once you take a vaccine, you expect your immune system to be able to be activated, either causing humoral aspect of the immune system to be activated or the cellular arm. And once you have the virus enter into your system or enter into the cell, then the humoral aspect of the immune system may be less of efficient to be able to clear the pathogen unless the T cells are activated. And the T cells, we have the T helper cells, the cytotoxic T cells, and then the Treg cells. T helper cells, we know, are able to coordinate other aspects of the T cell, like the CD8 T cells, that's the cytotoxic T cells, to be able to activate and then cause the necessary pathology that, or the necessary cell death apoptosis that we expect to see. So you have the T helper cells here. That is the T helper cell 1, 2, and then 17, with each of them having their activities to do. Now, once a CD8 T cell is activated, effector functions are also activated, where you have the cells causing, attacking or going into the infected cells and then causing the needed apoptosis. Now, the issue is that once these vaccines were available, there were still evidence of ongoing infections and also the mortality is increasing. So there was a need for us to get booster vaccines. And several reports have estimated the duration of this generated immunity from the COVID-19 vaccines, with some studies stating below 6 months, others stating 3 months, depending on the population. In Ghana also, we had our share. So this was a study conducted in Japan by Jinka et al. That stated that even though the vaccines were able to elicit the needed response, but the duration of the response was not up to 7 months, which means that you have to still get a booster to be able to have that kind of protection, long-term protection that you expect. In Ghana, we had also got our share of the boosters. And precisely, we have the Johnson and the Pfizer boosters that were administered among the Ghanaians. And our Ghana Health Service estimates that over 6 million people had received doses of this vaccine as boosters. But the question remains that even though the citizens or the population have received the vaccines, the immunological basis of this vaccine efficacy remains limited. The data concerning that basis of the vaccine efficacy, especially in the cellular aspect of immunity, remains limited. So the question that we intend to ask is, what is the longevity of this induced immune response that is conferred by the booster vaccine? And then what is the magnitude of the induced immune response in these vaccinated individuals? And does anybody who has already received a COVID-19 vaccine or have had any prior infection, do they have a higher chance of causing or developing T-cell exhaustion when they have received the booster shot? These questions are very important because it will give us the necessary information on how long one should receive booster. And even when you receive the booster, what will be the magnitude of response that you get, and whether your cells at a point in time will become exhausted or not. So this statement was that in Ghana we have received the vaccine, that is true, but then data concerning the immunological basis of it remains limited. So specifically, I was interested in looking at the cytokine stimulatory effect of two of the antigens, the S and N, in people who have already received the COVID booster vaccines, and by getting their PBMCs and then stimulating them. And also to assess some level of immune markers as markers of T-cell exhaustion within those recipients. And lastly, to compare between those who have received the Pfizer and the Janssen to see which of the vaccines did best in our local setting. So this was part of a longitudinal study funded by the Ghana Government COVID-19 Trust Fund to be able to assess the dynamics and then the immune responses within those who have received the booster vaccines in Ghana. I used 14 PBMCs from Pfizer and then Janssen vaccinated booster recipient. And we took samples, blood samples from them before they received the booster, 3 months after the booster, 6 months, and then 9 months after the booster to be able to assess the dynamics of their immune response. So we have concerned the participant collected our samples at month 0 or the baseline 3-6 months, and then each of them we isolated PBMCs and kept them in liquid nitrogen. To be able to assess the cytokine expression levels, we have to do stimulation, or I did stimulation, by towing the PBMCs and then stimulating them with the COVID antigens S and N. From there, we put it, or I kept it in the incubator for 48 hours and harvested the cell-spontanean to be able to run my luminous multiplex assay to determine this cytokine. These cytokines have actually been selected because, especially interferon gamma, TNF-alpha, interleukin-1 beta have a very key role in this antiviral immunity, and alongside interleukin-10 and then interleukin-6. This represents the demographic characteristics of the participants. So the mean age of the Janssen recipients, 27, and that of the Pfizer, 36. We also look at the gender and then the educational level in addition. Now the Janssen vaccine induced, so we looked at this is M0, that is before the booster administration, M3, M6, and then M9. So we looked at the interferon gamma expression levels across the time point. And we see that even to month 9, you see some significant levels of the interferon gamma cytokines being expressed here. This was also true for that of interleukin-1 beta and that of TNF-alpha. We looked at the Pfizer vaccine to also look at the expression pattern. At month 0, 3, 6, and even month 9, you see significant levels being expressed, some levels of quantities being expressed for interferon gamma, interleukin-1 beta, and then TNF-alpha. Now, we wanted to look at the impact of natural infection in these cytokines that are being secreted, and whether it was precisely due to the vaccine effect or it was due to natural infection. And for anybody to be able to detect the nucleocapsid, then it means that the person must have gotten a natural infection, since those vaccine designs were actually designed with the spike protein unit. So we checked the levels here across, and we saw some quantities expressed, but not as high as that of the Janssen and then the Pfizer in the spike protein. Now, in order to avoid cytokine storm, once immune system is activated, you expect to have some levels of immunoregulatory activity or immunoregulatory cytokines. So we looked at interleukin-10, its levels in both the Pfizer and then the Janssen, and we saw some significant levels in the Janssen recipient and also in the Pfizer recipients. But in the Pfizer, we saw the diminishing levels here as it goes to month 9. As it goes to month 9. Then the next thing is to look at these cytokines have been secreted, but is there any T cell exhaustion within the cells that were stimulated? So we looked at the cells after harvesting the spontaneant. We stain the cells, or I stain the cells with specialized antibodies, and these antibodies were for detection of PD-1, CTLA-4, TIM3, which are molecules that deplete T cell exhaustion, and also the chemokine receptor CSCR3 that gives indication of how active your T cells are migrated from a site, have migrated to the site of infection. So by staining this, I did acquisition with a flow cytometer at WACCBIP here to determine my immune markers. I saw low levels also expressed for both the CD8 PD1 and then CD8 CD1A3, CD8M3, and then CD8 CD152. PD1 This is team 3, and then CD152 to CTLA4. Low levels expressed for the Janssen recipient, and the same was also seen for that of the Pfizer recipients. The next was to do compare between the two vaccines to see which of them may have done better. Then we look at for the interleukin-1 beta at month 0, month 3, month 6, month 9. There's appreciable or comparable levels of both the Pfizer and the Janssen before the boosters, and then at month 3, month 6, and then month 9. This was also seen in the TNF alpha and then the month 3. But in the month 6, we saw the Janssen levels drop. Let's see, the Janssen levels having a little impact or little effect as compared to the Pfizer. This is also same for the interferon gamma levels. The next was to compare between the two vaccines, so the immunoregulatory cytokines. So we looked at the interleukin. This is before the boosters were administered, but at month 3, 6, and 9, we saw variations in the results, but it wasn't statistically significant. Then lastly, I compare between the immune checkpoint markers or T-cell exhaustive markers to see whether any of the vaccines had better secreted or had higher quantity of T-cell exhaustive markers expression than the other. But comparable levels were seen for the PD-1 across, and for team 3 and then CD152. But for the CD183, which is the CSCR3 at month 9, we saw the Pfizer having a higher or significant quantity expressed. And this CSCR3 is actually, it plays a key role in trafficking or in moving of T cells to the site of infection. So in concluding, the outcome of the study indicated or depicted an activated cellular immunity, specifically the T helper 1 immunity, in both the Pfizer and the Janssen vaccine recipients. And also the magnitude of the induced responses was comparable between the two vaccines. And we saw minima or little T cell exotic markers in the study participants. So I want to acknowledge the Ghana COVID-19 National Trust Fund for funding this project, and then the Noguchi Memorial Institute for Medical Research, where I work as a research assistant, for the opportunity given to me. Also my department, Medical Biochemistry, for the opportunity given to me to have my MPhil. And specifically with the WACCBIP flow cytometry unit, Dr. Danapra and Mr. Evans Niyama for their contribution. And lastly, I want to acknowledge my supervisors, Dr. Vincent Ama of University of Ghana Medical School for the contribution, and Professor Kojo Asamoah-Kwesi for his immense contribution towards my thesis and my whole education. Thank you.
Thank you very much, Mr. Frank Osei. Please, questions for Frank.
Okay, all right. Okay, thank you very much. Mine is just a comment, whether you looked at it during the design phase. It would be interesting to know the T-cell, again, what variation we're saying from the two vaccines.
Thank you. Yes. Thank you very much. I had a question. From your stimulation data, it looked as though there wasn't much difference between the pre-booster and post-booster. So when I was looking at your plots, I didn't see much of an increase between M0 and then M3, M6, and M9. So is that the case? Was there really no added, were the boosters not really helpful? Did you not see any difference in T-cell response between pre-boost and post?
Yeah. Okay, so for the cytokine data, like we rightly said, there wasn't any significant difference between the data generated. But then on other aspects of the vaccine also, we looked at the antibody responses that were induced, which is not part of this presentation yet. That could be discussed later on. And that we saw a significant shift at month 3, between month 0 and month 3, in the those who have received the booster, especially in the Pfizer recipients. So my take is that that's significant, but with the humoral aspect, that may be significant. Yes. Thank you.
Okay, I have a question for you, Mr. Frank. Yes. And my question is based on why were you working on?
In month 6, for the cytokine data, I could see consistently across different cytokines, there were 2 groups. And there were always two groups, those who were not responding at all or producing any cytokine, and those who were responding. That was happening in month 6. Even with stimulation, it seems to be the same. So I was checking, were they the same individuals that were not responding for all the cytokines in month 6? And if that was the case, were there any other confounding things that may, like other infections?
Yes, so you rightly said, in my CV saw low level expression. One, we didn't check as to whether they are the same individual that moved, that had that kind of drop. But this is what I have known from the other aspect of the work that we are doing with, especially with the humoral aspect, the antibody. Even at the month 6, there was a drop in the antibody levels. So what we are trying to do is to possibly look at if there was any variant that was possibly circulating within that month that may have affected the outcome of the results. And that is also another aspect of the work we are trying to do to find the answers to that drop in the month 6.
Thank you very much, Frank. Before we take our leave, I will break protocol a little by acknowledging the presence of my lecturer and later a colleague in the person of Dr. Ajimini. Dr. Ajimini, you are welcome. So this brings us to the end of this section. We will invite the sponsors to take over now. Thank you. Do you have anything?
No. Okay, I think that's it.
Thank you.
Yeah, thank you, Prof.
Let's thank Professor Kwashi Manfujira for a wonderful chairing of the session. They bought us some time. So, Andrew.
Yes. So, thank you once again. At this point, we wanted to give our benevolent sponsors some time to interact with us, but they have all agreed that we've done so much. We've already spent some time today, so they will rather start their interaction with us from tomorrow. But that notwithstanding, we still want to acknowledge our sponsors once more. Most of these are companies or organizations that deal with laboratory equipment and reagents, and we provide a platform for scientists to know where to procure some of their, scientists in Africa here to know where to procure their laboratory equipment and reagents from. So we have been fortunate enough to have, as platinum sponsors, LabMat and then Thermo Fisher Scientific. Then under the gold, yes, so LabMat and Thermo Fisher Scientific jointly came together as a platinum sponsor. Then we have Caramo, Yamachi Biotech, DCL Laboratories, Excelsior, and SRL coming under the gold category. Then silver, we have Nkaba, and then the University of Ghana Enterprises Limited, taking the silver spot, and then Bethel Logistics, or Betlock for short, coming on board as bronze sponsors. And also MES Equipment. MES Equipment is a Ghanaian-owned company. They also deal in laboratory equipment and reagents. They have been kind enough, and most of them, I think this year we are having for the first time Thermo Fisher and also SRL, as well as UGEL, University of Ghana Enterprises Limited, for the first time. Yeah, so apart from these ones I've mentioned, the rest have been very regular, and they've more or less become like a household name. So from tomorrow, we will create an opportunity for them to interact directly with you and give you presentations on some of the services they provide. Thank you very much.
Thank you, Andrew. And I believe most, if not all of them, have booths that we invite you to visit to see, especially those researchers here, to see what services that they provide. Okay, so we're going to move on with the schedule, and we're going to now have immune responses and disease pathogenesis, and we'll kick it off with our own Dr. Yao Bediako, and he's going to talk. But before, actually, I should have invited the chairs yet. Professor Kojo Kuram, Professor Kojo Asamakusi was supposed to be one of the chairs, but he's been called away, so I will chair in his stead, and I'll chair along with Henrietta Mensah Brown, Dr. Henrietta Mensah Brown. So, in my joint role as chair, I'll call Yao Bediako to give us the second plenary talk of the day. Yao.
Thank you very much. Speaking at WACCBIP is like speaking at home, so. But it's nice to see so many people, especially shout out to Jean Langhorn and Kevin Marsh, the two people who probably mentored my postdoctoral career and gave me a lot of opportunities. So it's nice that both Kevin and Jean are in the same room. So I'm going to talk about a project we've been working on. It's actually a collaboration between Yamachi and WACCBIP. You've probably heard a lot about Yamachi today, and a few people may not know about Yamachi. So I'd be reft if I didn't spend one slide or two telling you what Yamachi is. So Yamachi is a biotech startup that I co-founded. I currently act as the CEO. I'm also a research fellow at WACCBIP, but increasingly spend more and more of my time at Yamachi. And we are a life science startup interested in trying to understand cancer in particular among people of African descent. But that doesn't stop us from dabbling in a few other disease areas as well. So we started in 2021, began here in Accra, which is where we're headquartered. We've since built relationships in 9 African countries. We have a lab in Nairobi, and we have an office in DC. So we're gradually expanding our footprint, but we're very much a Ghanaian/African startup, and really born out of WACCBIP. I got my shot back in Ghana, returning to research, working under Prof. Awandare, and he was very supportive of letting me sort of spend some time trying to figure out if I could be an entrepreneur. But I think this is what WACCBIP stands for, is trying to allow scientists to branch out. And the goal is ultimately to enrich the ecosystem so that one day companies like Yamachi can support the work that happens at universities across Africa, so we're less dependent on overseas help. You can see some of the partners we're currently working with, including IQVIA, Roche, and a few others. This our lab is in Abelempe. You're all welcome to visit. A few of my staff are actually here, so now I'm nervous. But we also recruit heavily from WACCBIP, which is why I keep my links to WACCBIP, because I'm always looking for the best talent to recruit over. And these are the kind of things we've done in the last 3 years. We have a number of notable achievements, a number of publications coming out, and a number of grants and other projects we've been involved in. Now, one of our largest projects, some of the earliest grants I received as CEO of Yamachi, was actually a collaboration with WACCBIP. It was funded by the Bill and Melinda Gates Foundation through a Callistus Juma Fellowship that was awarded to me. And the goal was to understand COVID-19 immune responses among people of African descent or people across the African continent. We've since dubbed it the Heritage Study, and you can see our logo with an antibody and the Ghana flag. I think there's somebody here who is, Ida Menu in the front here is responsible for the logo. So, but anyway, it's quite an interesting study, and one that we have some exciting results from that I'll be sharing with you. It's a 5-year project. We're currently 2.5 years in, but we have some pretty cool results that I'll be very excited to show. And we also had great support from the Ghana Health Service, who allowed us to operate within their health system. So the main premise of Heritage is really to try to understand why vaccines seem to, demonstrate geographical differences in efficiency or efficacy. So if you look in the literature, especially for pediatric vaccines, you'll find there's a lot of talk about how vaccines seem to work really well in the Northern Hemisphere. They work well in Europe, North America. Bring them to Africa, and they don't quite work as well. Malaria vaccines, for instance, work really well in Oxford, and they're not quite as well here. Of course, that sometimes is due to the variance in lab generated malaria versus others. But when you look at norovirus and a few other vaccines that are commonly used, you see these differences in efficacy between Europe, North America, and Africa. And there's a whole number of hypotheses around why this happens, ranging from host genetics to environmental exposure to T-cell exhaustion from chronic infection. But of course, we'd like to be at a point where we can engineer vaccines to work well everywhere. Now, COVID-19 gave us a unique opportunity. It's the first time that the same vaccines were being used globally because we had this huge pandemic, we had a shortage of vaccines, and so all of a sudden you had a situation where people in Africa were getting Pfizer vaccines and AstraZeneca vaccines that were identical to what was being administered in the UK, for instance. And so we sprung upon this as a unique natural experiment to allow us to begin to tease apart why, if there would be differences in efficacy, and why these efficacy differences would exist. So the study we designed was initially a 12-month study of detailed characterization of a cohort of adults from Ghana who we would collect samples from at a pretty rigorous sort of time scale, ranging from before vaccination to 1, 3, 6 weeks, then over 3 months, 6 months, and 12 months. So a total of 7 time points, very, very carefully tried to chart the kinetics of immune responses who we recruited into the study and managed to follow for 12 months. So it's pretty large. Considering these were healthy people, we had to incentivize to join the study. And in the end, we collected way over 15,000 individual samples. And certain people in this room are responsible for a lot of that work. Now at Yamachi, we also build software, and clinical data is really difficult or really sometimes complicated to collect. This is just a plug to our engineering team if people are interested. So this is Uvosio, which is a tool we built at Yamachi that allows us to collect clinical data. And all the study data for Heritage was collected on our own platform. So it's, think of RedCap and Kobo Collect, but way better. At least we think so. So the data so far, these are the breakdown of the demographics of the participants. So 301 adults. You can see we had a bit of a skewing towards male. Many of them are in the age group 20 to 29. So we had a mixture of University of Ghana students with others in other areas of Accra. Employment status, majority were in the informal sector. So we did have some students, as you can see, but then we had a large number of people who were informally employed, and then some people who were sort of more office jobs in the formal sector. Most people had not reported any symptoms of COVID, which is interesting based on the data I will show you next. And then we had some with comorbidities. We had a small cohort of autoimmune disease people, so people with lupus and arthritis. And then we had a pretty good split between AstraZeneca and Pfizer vaccines. So at baseline, we PCR'd these people every time we sampled them. And at baseline, you see, although none of these people reported symptoms on the day, baseline is the day they were getting their first COVID vaccine, 20% of them were PCR positive for COVID. They were not sick, but when we did PCR, they were confirmed to be COVID positive. So these were all asymptomatic infections. And I can see over time, week 1, similar numbers, but then as we proceed after the vaccination, the proportion of people who will be PCR positive dropped significantly. And by month 12, nobody was PCR positive at the time we sampled. Of course, we may miss a few people, but what we did was we had weekly calls or bi-weekly calls where we would call people to ask how they were feeling and request them to come in if they had any symptoms, and they got a free complimentary COVID test at Yamachi. So we did as much as we could to really chart the COVID infection status across this population. This is just another fancy way that Yakubu, our statistician, helped to show their infectivity. You can see positivity. People go negative, then some of them are positive again. And then in general, we had this blip at month 6, but in general, a lot of people remain negative. And if you're wondering, obviously, we can't recruit 301 people in one day, so we did it in batches. So the first baseline recruitment was done over a period of a few weeks and so on, and then we just followed them up at different intervals. And you can see our COVID positivity lines up very well with the Ghana infection data. So as peaks emerged in Ghana, our cohorts mimicked what was happening in the country. So we think our sample was pretty representative of what was happening in the country. You can see we had a little bit of a peak in June 2022. So we started off in December 2021, which was really the Delta wave, then June, or Omicron, sorry, and Peter is the expert. And then we had a little bit of blip here. Now this is vaccination status, so you can see that Ghana was vaccinating, but it wasn't very rapid over that period. If you compare that with global numbers, you can see globally vaccination rates were higher, but then there was a global blip in December 2022, which we didn't see in Ghana in our cohort also did not reveal, which is quite interesting. Now, if you want to look at a bit of multivariate analysis about what kind of stood out, we found that participants were more likely to be positive within the first 3 weeks post-vaccination, not surprising because they were unvaccinated. After they got the vaccine later on, they were less likely to be positive. So, the vaccine seemed to be working, which is nice. And Ghana Health Service likes to point to this data, so they were happy to work with us. When we look at comorbidities, people with comorbidities were more likely to be positive at the time of vaccination. Once again, makes sense. Interestingly, people in the formal sector, so people with office jobs who worked indoors, were more likely to be positive than people outside. Now, there are two possible explanations. We can get into this. One is that, well, if you work outside, you're fresh air, you're not likely to breathe in COVID in a closed environment. The other alternative is people outside had got COVID a long time ago. Their antibodies levels were really high, and the bougie people in the offices were now getting hit. And I'll show you some evidence why I think that might actually be the case. And then we had interestingly, our autoimmune group was less likely to be positive. Now, we think that points to health-seeking behavior. If you know you have lupus, you know you have an autoimmune condition, you probably take the mask wearing and those mandates a bit more seriously than others. Now, we were interested to understand, we knew what people reported as, but somebody can say, I've never had COVID and maybe just been asymptomatic. So we did, this actually work we did with the CRIC, looking at serum level anti-spike and nucleocapsid. Peter's group has also done similar work here in Accra, but these samples were run at the CRIC. And so yes, I showed you 20% COVID of SARS-CoV-2. So wild type is your Wuhan strain. This is baseline all the way to month 12. Our individuals maintain pretty high neutralizing ability over 12 months. And this is in the absence of boosters. None of our, or very few, in fact, hardly any of our participants had a booster in this 12-month period. So even in the absence of boosting, we maintain pretty high levels of neutralizing antibodies, even against variants that were very different from the wild type. These people were given Wuhan strain vaccines. Ghana didn't, most African countries did not benefit from the modified, improved vaccine boosters that were available. So even in month, and interestingly at baseline, XBB did not exist when the baseline samples were taken. And yet you can see that even at baseline, you have pretty good neutralizing ability against a future variant, suggesting sort of some cross strain protection. Now, the beauty of Heritage is we have Legacy to compare it to. Legacy is a CRIC study that is looking at UK adults. And so the design of Heritage was to mimic Legacy to allow us to now compare, to answer our question about Africa versus Europe, to really compare a similar cohort. So we extracted a matched group of people who had similar times to vaccination from Legacy and compared them to our Heritage cohort. And what you can see, if you compare this reference dotted line, straight away, the African or the Ghanaian participants had much higher neutralization levels than the UK counterparts. In fact, for all the variants, and especially as you get into the Omicrons, the levels of neutralization ability drop significantly. Now, these groups had received some boosters. So the interesting thing is for the later time points, even boosted UK participants did not match the level of neutralization that we saw in the unboosted Ghanaian population. And you can see the time series. Legacy is a slightly older cohort, so they started in January, right when vaccines became available in the UK. We started in December, right when vaccines became available in Ghana. We don't have to get into, I'm just trying to trigger you a little bit. But anyway, these groups were matched, though, in terms of time to vaccine. So as much as we could, we've matched these groups, and I think these differences are quite striking. And you can see them even better here when you do side by side at the different time points, the different variants, and then this is Heritage and Legacy. You can see straight away there appears to be quite a difference, especially when you get to Delta at baseline. So at baseline, before any vaccine, our participants were neutralizing Delta very well. Of course, Delta was all over Accra at that point, versus Legacy patients who were a year earlier before Delta came, and as you go down. Now, we asked ourselves, is it just that the Ghanaians were just super? I mean, we know we're super, but we wanted to be slightly more scientific. So we thought, what could explain these differences? So remember this group down here. Remember I said there were a group of Ghanaians who were truly negative, at least based on ELISA of their nucleocapsid. So they had somehow managed to be nucleocapsid negative in the midst of 80 to 90% seroprevalence in Accra. And so we thought, let's compare these two groups. So let's break down our heritage cohort according to the nucleocapsid positive at baseline versus nucleocapsid negative at baseline. When we do that, a very interesting thing emerges. All of a sudden, our nucleocapsid negatives look much lower. So they have much lower neutralization ability relative to the nucleocapsid positives. And this is maintained for all the variants. And you can see for Omicron, which was around the time, right, around the, which was around when we began this study, you can see that in 12 months, you finally reach the nucleocapsid negative catch up with the nucleocapsid positive. But for the other variants, the historical variants, Wuhan, A27, and Eta, which were no longer circulating, the nucleocapsid negatives never catch up with the nucleocapsid positive. So we think this is really beginning to tease apart the role of exposure and continuing exposure in the Ghanaian cohort with neutralization levels in the context of vaccination. And then when we bring legacy back into the mix, all of a sudden legacy almost overlaps with our nucleocapsid negative. So it sort of begins to move away from some sort of genetic difference between the Africans and Europeans, and rather the fact that the UK cohort was more negative at the time they got their vaccine, whereas the Ghanaian cohort was largely already pre-exposed. So you had a certain amount of pre-immunization or a certain amount of pre-exposure, and then the vaccine came on. So the vaccine actually acted as a booster as opposed to a primary response. Whereas in the nucleocapsid negatives in Ghana, they got the vaccine as a primary response and then took a while to then gradually ramp up. Of course, also being environmentally exposed beyond that point. So I think it's a really important point to show that in the case of in Ghana and in many African countries where vaccines were delayed, in this case, it actually became almost a prime boost approach where environmentally we were primed, vaccines came in. Vaccines helped us because you see this significant rise, so the vaccine was useful. It's good we got vaccinated. In some ways, actually, when we got vaccinated, it helped us even more, and we're able to maintain this level. Now, this maintenance may be due to exposure, and we're teasing into that with longer time series now. But I thought this was really quite a striking result, and one that hasn't really been published yet. So we're working very hard to get this out in the next month or so. So what we're doing now, we have all those wonderful samples. The next part of Heritage is to now dive into mechanism. So we're looking at T-cell response, we're going to look at transcriptomic responses, and now we kind of have the groups to compare to, right? Now we have our nucleocapsid negatives versus nucleocapsid positives. We're going to use that information to now understand how to begin to tease apart the mechanism. And of course, we will also have our match cohort and the legacy group that we will continue to compare to. And the great thing is good work leads to more good work. And we joined up with WACCBIP to apply for a welcome grant that we were awarded in partnership with CRIC again, and this time also incorporating University of the West Indies. And so now we're doing the WWW consortium, which is like West London, West Africa, and the West Indies. And the idea is to now look at Caribbean populations who are genetically similar to West African, but environmental exposure levels were more similar to Europe than in Africa. And so this allows us to begin to tease apart even more this difference that we're beginning to see. So this is an add-on which allows us to now follow our heritage cohort for 2 more years. So now we're going to have that downstream, 24 and 40 years, really to see whether or not these protection are maintained as environmental COVID has begun to drop. So I know I'm at the end of my time. I just wanted to highlight this. Kese gave a nice little 5-minute talk earlier on. But Kese is a student co-supervised by Peter. And really, the big thing we spoke about there was the link between asymptomatic COVID-19 and recent malaria exposure. And the idea that malaria exposure may actually prime the immune system to be a little bit more immunomodulatory, a little less inflammatory, which may actually end up being protective or at least helpful in the case of severe COVID. So that is out. You can check it out, malaria reactivity. So this is, I'd just like to start, first of all, thank the study participants, 301 Ghanaian healthy adults who chose to join up for Heritage. About 200 of them, or 150, 180, I think, or 160, right? So anyway, around 150 have stayed on, so we're following them for 2 more years, which is probably a tribute to their stick-to-itness and our team's stick-to-itness as well. This team at Yamaha. Oh, I knew Kevin would have a question.
Okay, thank you very much, Yao, for that wonderful presentation. We have time for just one question, because Yao spoke over time. So I'll let Kevin go ahead and ask his question.
Can you hear me okay?
Yeah, okay.
That was really great.
so much data that you probably shared this and I just missed it, but one big difference, of course, or the biggest difference between a Ghanaian population and a UK population is in the age structure. Now, when you compare the two things, you obviously you match them, so you'd have matched on age. But when you look at the unmatched data, are there any obvious age-related differences? Because so you're getting kind of, if you like, you're saying it prime boosting, but prime boosting in a young population may be very different from prime boosting in an old population. So I just wondered if you'd like to comment on any of those differences. So yes, of course. I mean, actually, I think the legacy cohort is derived largely from the quick start, who themselves probably don't very well reflect the UK's age. It's probably skewed towards PhD students, postdocs, and some PI. So it's probably a spread. So we do have a few slightly older people in our cohort, but our cohort is, as you saw from the data, largely 20 to 29. So that is a good point, that prime boosting might be slightly different. But our cohorts are matched. So in this case, there shouldn't be a difference, but it would be interesting to see when we take those extremes, whether we would. I answered quickly, so maybe I can.
Okay, I think, okay, we can take
one quick one.
Two questions from, one from last.
Positive as a baseline compared to those that was not positive. Thank you.
Okay, so I think your first question was about SCT values. They range, but somewhere as high as 14 or 15. So these people had pretty high, viral loads, but were asymptomatic. Your second question, I wasn't sure if I caught you. You were talking about whether we saw differences in the vaccine. Yes. Oh, so that is a good, whether those who were positive at the day they got the jab had differences in reactivity. So that is something we are beginning to tease out. We'll probably have a better chance looking when we start storing the PBMCs. Right now, the data we have is serology, where we didn't see any difference. But it's unlikely you see a huge difference from antibody responses. It's unlikely you see a huge difference from antibody responses from that group. But that is something we're going to look at when we start pulling out the cellular. Lars.
Small question related to Clemens, actually. The small group that you had that were initially protein negative, are they age do they have the same age as those that were positive, or were they younger or older? Do you remember?
That's a good point. I'd have to go and look in the data. I don't think we've looked at that.
Otherwise it might
Yes.
be important because otherwise
Yeah.
you can't really compare them.
Yeah. So we'll look. I mean, we didn't have any children, so everybody was 18 or above. But yes, it would be. Yeah.
I mean, difference
Yeah.
between an 18-year-old and an 80-year-old.
Yeah, we didn't have any of those either. But I get your point. Yeah. So I know. I think it's good that we'll go back and look.
Increasingly mindful of that difference.
Okay. I won't ask why.
All right. I think we'll continue those discussions later. And we're actually back on track. Thank you, Yao. So we'll call up the first of our fellow session, Edwin Magomeri. And he's going to give us a talk on HIV-1 and HIV-2 interaction reduces cell infectivity and viral production in vitro. You have 12 minutes and for your presentation and 3 minutes for questions.
Okay, thank you. So I'm Edwin Magomere, and my talk is on HIV-1, HIV-2 interaction reduces cell infectivity and viral replication in vitro. I'm a PhD fellow at WACCBIP. And so over the last 4 decades, we have seen tremendous progress in HIV, especially when you talk about drugs and treatment and trying to achieve cure. We have some of the best ARTs that can really suppress the virus. However, looking at the numbers on the screen, you see that over close to 40 million people are HIV infected currently. We also have new infections which are still going on. And therefore, this calls for a concerted effort towards getting a new cure for this HIV. And just a little about HIV. This is a virus. It's a very tiny virus. Its full name is human immunodeficiency virus, and it's classified under the genus lentivirus. It is a retroviridae, and there are 2 major types of HIV. The first type is type 1, which is grouped into 4 different groups. These 4 different groups, only group M has been shown to be relevant in terms of epidemics and disease progression. So this group M normally has many different clades, clade A, B, C, and the rest. And when individuals are infected with more than one clade, they result to formation of what we call circulating recombinant forms. For type 2 HIV, normally it's grouped into 8 different groups. Just looking at the snapshot of the map of Africa, we see that HIV type 1 is prevalent across Africa in most regions and almost all the countries. However, take note of the different clades that are driving epidemics in different parts of the continent. On the flip side, we see that HIV type 2 is mostly confined to West Africa. I will not bore you with the life cycle of the virus, but I want you to take note of this. This is a tree. I'm trying to use this to illustrate how the virus replicate and how we have taken advantage of the different replication cycle of the stages to develop different drugs. So consider the roots as the process when the virus gets entry into the cell, then the trunk or the stem as the RNA replication and transcription steps, and therefore the virus normally has to make sure that the RNA has been translated into different viral proteins which must be assembled into a new virus. Looking at this tree, you see that most of the current drugs are targeting the leaves which are representing the different HIV proteins, and we have not taken given much attention to the most important part, which is the transcription step and also the cell entry. Although we have some of the vaccines and drugs targeting this step, but they have not been able to cure the disease completely. So the question we are asking, if we have specific HIV-specific transcription inhibitors, will they be able to stop the process from this point? So this rationale of this work, back in 1993, this work was published showing that individuals who are infected with HIV type 2 have some level of protection against getting HIV type 1 infection. Subsequent studies have also shown that when individuals are infected with both type 1 and type 2, their progression, the disease phenotype is normally reduced. They progress more slowly. Their asymptomatic stage is a bit more prolonged compared to those who have only HIV type 1. So what we are trying to understand, is it an antagonism between these two viruses when they coexist in the same cell? And if we are able to understand, if you are able to understand this interaction, we can actually gain some therapeutically useful insights that can help us in development of new drugs. So what the problem we are looking at is that HIV is here with us and is killing, despite the fact that we have drugs. These drugs are not safe. Most of them are not scalable to a large scale. The drugs, also the virus is also very clever. It develops resistance against the drugs that we have. We also have the ability of the virus to establish a latent reservoir, and we don't have the vaccine for this virus. Also, with the presence of HIV type 2, especially in western part of the African region, is something to worry about. So we sought to assess the viral infectivity in single and dual infections. Then we also sought to evaluate the effect of dual infections in HIV-1 productive infection. So just to do this, we started with seeding our cells. Our cells of interest are U87 cells, which actually are susceptible to HIV infection. Then we had to do this over 24 hours to allow the cells to confluent. Then after that, we did infection with either HIV type 1 or type 2. After that, the viruses that we used were kindly donated by the National Institute of Health AIDS reagent program. Moving forward, after about 96 hours, we harvested our viruses, and after harvesting the virus, you have to quantify and know the quantity of the virus that is present, which is actually infective. So we did the first method, which is crystal violet staining. Then we had luciferase assay, which we used to quantify our viruses. Then thirdly, we did a P24 assay to just confirm whether the virus were actually present in the cell supernatant. So moving forward, I'm showing you the first image. The first image, which is is an image of infected cells. You can see, it's not very clear here, but you can see lumps of cells, masses of cells, which the arrow is pointing at. This is an formation of a syncytium, which happens normally when cells are infected with HIV. This is at day 4. Then at day 4 again, we have, at day 4 again, we also have cells which are not infected, and you can see that there are no formation of syncytiums. So this table here gives us the TCID50 that we were able to calculate using this formula here. And if you look at the TCID50, these are the tissue culture infective doses that can kill 50% of the cells. And we realized that most of them were into their thousands, apart from this one, which was about tens of thousands. So we wanted to confirm whether our TCID50s were actually effective, and we used the one with the highest TCID values, and then with the moderate TCID values, we did a serial dilutions at MOI of 0.5, and you can see the curve that we were able to generate showing that indeed our TCID50s were okay. So having known that, having established the TCID values of our viruses, we went on to look at how does dual infections affect viral infectivity. So the method here shows us how we were doing, we used this cell line TZM blue, which is susceptible to infection and also can be used for luminescence assay. So this is infection for 48 hours. After 48 hours, we harvested the cells and did our luciferase assay. We also went down to extract RNA from these cells and quantified unspliced HIV RNA as well as multiply spliced HIV RNA. Then we also extracted RNA from the supernatant, where we were able to perform viral load to confirm how much of the virus was actually released into the supernatant. So looking at the data, we see that when we perform a simultaneous infection is when you introduce both 1 and 2 at the same time. We see that there's quite a significant reduction across. So this first one is HIV-1 alone. This is HIV-1 plus HIV-2, then thirdly is HIV-1. Across all the graph, you can see some reduction in terms of infectivity. And so we wanted to know, is this something that's just happening be sustained over a period of time? So we were able to do this at 24 hours, 48 hours, 72 hours, and 96 hours, and we established that for all the single infections represented in blue, we see there is a higher infectivity compared to the dual infections, which, sorry. Thank you. So compared to the dual infections, which you see lagging behind, which you can see they're lagging behind the single infections, then this is HIV type 2 infections for over a period of 4 days. Our findings were not unique. They have been reported before in literature where this work was done using GFP-tagged viruses infected in single and dual infections, and have a cell measurement done after about pictures. So having established this, we wanted to know the initial one was the simultaneous infection, but what if we do a sequential infection where we start with type 1, then type 2, or we can reverse the order from we do type 2, then type 1. What we saw with this one is that when we did a sequential infection, there is a slight reduction in terms of infectivity between the single and dual infections. And this was when we began with HIV type 2, then HIV type 1 later. When we reversed the order of infection, we saw that the effect was actually lost. When we reversed, we started with HIV type 1 first, we see that there is the dual infections are not really reduced as compared to those which were done when HIV type 2 was put in first. So again, we wanted to see how these dual infections would affect viral production of viral RNA, that is unspliced HIV viral RNA, as well as multiply spliced HIV viral RNA. So we see that when we do a simultaneous infection, we see that there is reduction. This is relative, this is the expression of unspliced RNA relative to single infection. So take this graph as if the 0.0 is where single infection is, then we are going to, the expression levels for the dual infections are going to the negative values, which shows that there is a reduction or rather a down regulation. When we did a subsequent infection, starting with HIV-1, HIV-2, then HIV type 1 later, we see that there is reduction in 3 of the clades we used, apart from one clade, the clade B, which was showing an increase in expression of the unspliced HIV RNA. So this, again, we saw in literature has been done before, and an explanation to this is that normally when HIV type 1 and type 2 coexist in the same cell, they both require TAT or TAT for transcription of their RNA. So when type 2 is present, it's been shown that the promoter for type 2 is has a high affinity to TAT, and therefore making it unavailable for HIV type 1 to continue with its transcription. This study here was done again back in 2019, and they showed that when you use transcription inhibitors, this is a transcription inhibitor, dihydrocortistatin A, it also reduced a level of infectivity in cells. So we wanted to see if the cell-associated multiply spliced. This is in effort to try and understand if this dual infection would affect splicing of the viral RNA. So we see that here, when you do simultaneous infection again, 3 of the viruses were reduced, while only 1 showed an increase in their levels. So for the subsequent infection, we didn't see a clear trend. So it's something that we might have to repeat again. So just to cap it up, we wanted to see if this dual infection would affect the viral load for all the subtypes we use, or rather the clades we used. So we quantified the viral loads in the supernatant, not in the cell, the supernatant of single infections and dual infections. Across, we see a reduction in viral loads. So quickly, what we are saying is that when there is a dual infection, we see a reduction in infectivity. Also, we see a reduction in transcription. Both of these work to result to low viremia, which is a good disease phenotype or other disease prognosis marker for HIV infection. But if you can remember our tree that I used before, if we focus our target on reducing infectivity and reducing transcription, is it possible we can make this tree die? Thank you. These are my supervisors and the funding bodies, WACCBIP, World Bank, and Francis Crick. Yes, thank you.
All right. Thank you, Edwin. We have time for a few questions. Sebastian.
That's a nice presentation.
Thank you.
What is that? Well, let me just start this way. So we know that the sequence is important, right?
Yes.
What is the therapeutic relevance?
The therapeutic relevance?
Yes.
Of the sequence?
My question?
Yeah.
Because you have a sequence.
Mm-hmm.
So how relevant is the sequence?
The order of infection.
Yeah, in therapy.
So the order of infection in therapy. Okay, so the reason why, first of all, I did the order of infection is naturally people can be infected with type 1 first, then type 2 comes in. Also, you can have type 2 coming in first, then type 1. Or one can also get infected with someone who is dually infected, so they can get both of them at once. So in terms of therapy, what it shows us is that even if we've seen an effect of HIV type 2 against type 1, in terms of reducing its infectivity, it's possible that when type 1 comes in first, the type 2 may not have really a significance in terms of reducing type 1. So when we try to mimic these steps, the 2 steps I have highlighted, the infectivity and transcription, we need to choose our targets carefully so that we can get a target that can actually work for both when we have both initial HIV-1 infection and then type 2. So it's something that we have to consider when we are choosing therapeutics or when we are developing new drugs. Yes.
Does that answer your question?
Doesn't. Maybe we'll. Okay.
Maybe you discuss a little bit after.
Yeah.
There's a question in the back, Darius.
Oh, thank you so much. So my simple question will be why you consistently will only select for CCR5 positive viruses, whereas is there a reason why you are leaving out the X-Force, which can be responsible for mixed tropic or dual tropic viruses?
Okay, yeah, because we actually knew the tropism of our virus, the virus that we are using. So that's why we went for CCR5. But we also have cells for XR4, which we can also consider later. So that will be better if we are using patient isolates. Yeah, then we can use both cell lines. Yes.
Okay. I believe Alfred has a question.
The restriction of HIV-1 by HIV-2, does this depend on the subtype at all because there are several different subtypes of HIV2?
Yes, the restriction could be subtype dependent because that's why we use 4 different clades so that we can see. And for especially clade B, it was not being really affected, but the clade A, clade C, and then the CRF-AG, we see a significant effect. But for clade B, we don't see a lot of reduction.
So at population level, which of the subtypes in HIV2 is more prevalent?
For progression?
Yes.
In terms of progression, we don't have that data yet.
No,
because you can get that when you are doing a cohort follow-up over time. But this is based on cell line work. Yes.
All right. So let's thank Edwin once again.
So we'll move on to our next speaker, Ebenezer Taylor. So Ebenezer will be presenting on a plasmodium-encoded nuclear distribution C-like protein is essential for parasite transmission. Ebenezer, you have 12 minutes. Thank you.
Thank you. All right, thank you for this opportunity. I think I will digress a bit from the normal human malaria to the animal models. So my name is Ebenezer, and I'm going to talk about the role of nuclear distribution C proteins in the malaria parasite in terms of the transmission stages. So I wouldn't bore you with the whole life cycle, but I will dwell on the stages in the mosquito. When in the surgeon stages in the blood, when it ruptures and releases merozoites, some are committed to sexual development. And so we have our male gametocytes and female gametocytes being picked up during a blood feed by the mosquito. The male gametocyte and the female gametocyte inside the mosquito midgut are under conducive environments in the mosquito midgut, such as a drop in pH, activates these male gametocytes to undergo 3 rounds of DNA replication, leading to formation of 8 male flagellated gametes. Whilst the female gametocytes undergo also activation, which runs up, and then these two will fuse up to form a zygote. The zygote undergoes transformation, leading to the formation of motile O-kinates. These O-kinates then migrate through the midgut and get encysted beneath the lamina of the mosquito, and then develops into an oocyst. This oocyst then matures and produces several hundreds of sporozoids that get out of the oocyst and then find themselves in the hemolymph, then eventually inside the salivary glands, which will then end up in the humans or the vertebrate upon mosquito getting another bite. So what is important in this parasite life cycle stages is that the malaria parasite life cycle is a bit complex, but there are 4 distinct stages where the parasite uses to multiply itself into several parasites. And these stages are very important, and they serve as potential targets for malaria drug development. Now, as a parasite divides, the nuclei has to move, and during the movements, like it moves during, during nuclear division in mitosis, the nuclei will move into daughter cells. Also, during pronuclear fusion, during fertilization, there's movement, just like in other eukaryotic cells. Also, there's position of the nuclei during interphase. These are studies that are found in other eukaryotic cells. Now, nuclear movement is an essential process that is vital for the cell growth and the development of eukaryotic cells. But these processes are regulated by several proteins in eukaryotic cells, such as motor proteins, microtubules, and then some coupling proteins like the dynactin. Aside these, there are important genes that code for genes known as the nude genes. These nude genes have been found to be essential in regulating microtubule dynamics, cytokinesis, mitosis, and other fundamental cellular processes in eukaryotic cells. Now, sorry. So, these new genes were first discovered in Aspergillus nudulans. And what happened is that it was discovered during genetic screening of mitotic mutants of this fungal species. And it was found that deletions of these new genes resulted in abnormal nuclear distribution in the fungi. So this is the wild type, and we found that in the wild type there is nuclear migration proceed smoothly compared to the mutant, which we found out that there is aggregation of the nuclei, means that they are not able to move. Now, so far, about 7 of these nude genes, which codes for nude proteins, have been discovered. And among them, the nude proteins seems to be very important in the fact that they are structurally and functionally diverse. And in humans or in mammals, about 4 homologs of these nude proteins have been discovered. However, despite the important role that these proteins play in other eukaryotic cells, the role of these proteins in malaria parasite remain largely unknown. So, my first strategy was first to search for presence of these nude C proteins in the malaria parasite. I mean, by doing blood search in the Plasmodium genome database and also in other epicomplexan parasite database. And it's clear that all 4 homologs of nude proteins are conserved across the epicomplexan parasites. So, for the purpose of my study, I focused on the nude-L of the Plasmodium parasite. So I called it as PB nude-L. The reason is that this nude-L has been found to play diverse roles, such as regulation of microtubules, dynamics, nuclear migration, cytokinesis, and even mitosis. So it's clearly an important protein in eukaryotic cells. I did a bit of homology analysis, and it shows that the PB nude-L, in terms of structure homology, it's almost similar to the human nude-L, and the fact that they all contain the N-terminal nude domain and the what they call the nuclear movement domain, which contains the CS domain. So now, to first look at the function of this protein in the parasite, I decided to determine the expression of the protein in the various stages of the parasite. So to do that, I just carried out what we call the single crossover homologous recombination. That is, I just placed GFP tag in front of my gene, and then so I generated what we call GFP construct of my gene, and then try to so after I've generated my constructs, I carried out transfection, and then but before that I needed to confirm the integration of my GFP tag in my parasite line, and then I did what we call genotyping PCR. So this PCR shows the GFP tag of my parasite line. Next, I use Western blot to confirm actually if this protein is expressed in the parasite. So I use Chargon pellets and then did Western blots, and this shows that the protein is expressed. I then proceeded to look at which stages of the parasite using live cell imaging to determine whether they are expressed in all the stages. And so the image over here shows that my protein is expressed in both blast stages and the mosquito stages of the parasite. But of course, there is no specific location. It seems to be cytosolic more of expressed. Since NuCl is associated with microtubules, I decided to co-stain with tubulins to find out if it's associated with tubulins. So from this result, it looks like the PB NuCl is actually partially overlapped with tubulin. So now I needed to carry out mosquito stage characterization to actually characterize the function of my gene. And so to do that, I mean, upon 2 attempts to knock out the gene, it failed. All right, and so it indicates that this gene is actually essential for blast stage development of the parasite. And so I resorted to what we call the promoter swap. And in the promoter swap, what I did is that I placed, how do you call it, a Clark promoter. I swapped it with the natural promoter of the gene. The Clark promoter will allow me to bypass the blast stage so that I'll be able to study the function of the protein in the mosquito stages. So I generated what we called P-Clark NuC lines, and then confirmed this by genotyping PCR over here. So compared to my wild type, and then the primers I used, it shows that I was able to generate parasite lines that contain my Clark promoter upstream of my gene of interest. The next was to actually see if indeed there is downregulation of my gene in the mosquito stages. So for here, we can see that the gene is expressed, with the GFP, it's expressed in the blast stage, but it's absent in the sporozoid stage. As we can see, there's no GFP signal in the sporozoid stage of the malaria parasite. So having successfully generated a parasite line of PBNuCl using promoter swap, I then proceeded to then determine the impact of this gene on mosquito midgut colonization and then oocyst development. So what I found is that compared to my wild type, there was decrease in infection rates, and also the oocyst numbers were lower compared to the wild type, and the difference were actually significant, statistically significant. As we can see from the prevalence actually is the infection rate. So there was about 83% infection in the wild type compared to my promoter swap lines. Next, I wanted to actually also find out if this knockdown of this gene could have any impact on sporozoid formation and then gliding motility of the sporozoids. So in terms of sporozoids, I determined sporozoids numbers in salivary glands and then the midgut sporozoids. So as shown over here, compared to the wild type, I saw, reduced sporozoids numbers in both the midgut and salivary glands of my swap lines compared to the wild type. Also, I then proceeded to look at if this affects the moving pattern of the sporozoids. And so we can see that the gene has an effect on the motility pattern of the sporozoids. As we can see, the normal movement of sporozoids is termed as gliding. So we can see that compared to the wild type, there's fewer of my sporozoids which glide normally compared to the wild type. So my final experiment involved transmission experiment. Here I did 2 experiments. I needed to actually find out if the gene being knocked down could affect the parasite or the parasite infecting mouse. So the first transmission experiment was IV, where I injected around 1,000 salivary gland sporozoids into mouse via IV. And so then I monitored the survivor and the parastemia. So there was decreased parastemia in my experimental group compared to the wild type. And also in terms of survival rates, actually there was no much difference because almost all the mice in my experimental group succumbed to infection. However, when I compared it to the natural infection mimicking, that is, I allowed the infected mosquito to bite or to feed so that they can transmit sporozoids into the mouse. So I found out that there was actually, once again, there was a decreased mean parastemia in my experimental group compared to the control group. And also, I mean, only 2 out of the 8 mice in my experimental group actually succumbed to the infection. And then indicating that the gene has an impact, or this protein has an effect on what you call transmission of malaria parasite. So in summary, I would say that nuclear distribution C proteins are highly conserved in eukaryotes and play several roles in the malaria parasite as well as in other eukaryotes. In this study, what I've been able to establish is that Plasmodium encoded NuCl is essential for transmission of malaria parasites. However, these proteins, the functions in the malaria parasites are not known. In other studies, in other model organisms, in other eukaryotes, they are known to play diverse roles. So it is important that these proteins are studied and their functions properly elucidated in the malaria parasite. So in conclusion, or as a form of acknowledgement, I would like to acknowledge these important personalities who have supported me so far, as well as these institutions. Thank you very much.
All right. Thank you very much, Ebenezer. We have time for one question, if there's any. One. Okay, Dave.
Thanks. That was a great presentation. Thanks. Question is a very simple one. You focused on that one protein. What about the other 3?
Yes, I didn't investigate them. I was focusing on only one. Yeah, I can't do. There are lots. Yes.
Right. If there are no more questions, we would like to thank everybody, thank all the speakers for this during this session. It's been a wonderful session. And then I think we have a few housekeeping announcements. We'll be going for a coffee break now. So the coffee break is just outside the WACCBIP building. If you use the door to the WACCBIP building, you turn left towards the car park. You can't hear?
Okay. Let me. I'll do it.
Okay. Peter will talk over there so you can hear. All right. Thank you very much.
So, am I Dr. Jekyll role or? Yeah. So we have coffee break now. It's gonna be 15 minutes, and you just go out the front WACCBIP door, go to the left, and for those who are here for lunch, it's exactly where lunch was. For those who have been here before, it's exactly where it usually is. We've caught, yes, and we've caught up on time. You know, people are not sure how we did it, but we always do it. So let's go for coffee break, but come back in 15 minutes. And while you're there, we have booths for our sponsors, and we also have some of these beautiful WACCBIP shirts that you can get. So see you here back at 5:10 for the last session of the day. All right, let's start settling down. When we fall behind in the beginning, we can catch up, but falling behind late, it's hard to catch up. Is Dorothy Obuobi here? Okay. What about Omikunle Falat? I believe that should be online. And that would be very interesting, especially the last one. Effect of toilet flushing. Yeah, the plenary is here. Yeah, he's here. You want to start? Jean, last year was 10 minutes. Yeah, that's why it's 15 minutes this time. But yeah. Okay, so I think we're gonna move on. Our plenary speakers here, our chairs are here. So before I'll introduce Reverend Dr. Winifred Seth Bewonyo and Dr. Anastasia Akins, who will be chairing this last session. And as if you noticed, this today has been primarily devoted to host pathogen interactions and immune responses and disease pathogenesis. So that's the theme that will continue throughout this session. So will the chairs please take their seats, and then we'll start this session. And this is the last session for the day.
Okay. Good evening and welcome to the last session of today's program. So we have one person giving us the plenary talk, and he's in the person of Dennis Pedro Calado. He's going to talk to us about modeling the development of endemic Burkitt's lymphoma in mice. Welcome.
Okay, so it's a real pleasure to share with you some ongoing work of the lab, and this is work in collaboration with Jean Lamborn, which I'm sure you all know, and the work done by a postdoc called Faith Uwaidia. And of course, I would like to thank the organizers for the opportunity to just share with you some of the data. So this is ongoing work, so any comments will be appreciated. So my lab specializes in understanding a stage of B-cell development called germinal center B-cell reaction stage. And as you can see, so B-cells develop in the bone marrow, so through a quite complex process that leads to the formation of this B-cell receptor on their surface. And these cells go on to the periphery, like spleen and lymph nodes, and becoming mature naive cells, which are quiescent in nature. But during infection process, like virus and bacteria, they enter and they form a germinal center reaction stage where actually they are hyperproliferative stage. And at this stage of the reaction, the cells actually change their B cell receptor, not in terms of their DNA itself, but they introduce mutations in the B cell receptor that was there before, and this goes through a process of selection to best receptors that recognize the antigen of the bacteria or infectious agents. And this ends up in the cells forming plasma cells, which are antibody-producing cells, or memory B cells, which are important for the recall responses. And we normally see and look at germinal center reactions by flow cytometry, but also by histology. And you can see here germinal center reaction B cells in the B cell follicle, so which is in blue, and adjacent to the T cell area. So this is just a picture of the spleen. So in the lab, and as Abdul also showed, so we are very interested in doing also more in-depth imaging, so we can actually now look at lymph nodes and see these germinal center reactions in the whole tissue. So, and you can see here represented in red by extending of BCL6, which is transcription factor, and you can see now these germinal center reactions taking place in the organ tissue. And you can see that there are quite a lot of them. So my lab is interested in physiology, so these processes of response to infection, production of plasma cells, memory B cells, these dynamics. But we are also very much interested in processes of pathology, such as autoimmunity, and related to what I'm going to talk about is the development of cancer. So both lymphomas and actually multiple myeloma, which are in both cases derived from cells that have either at the germinal center stage or have passed the germinal center reaction. So, and this is the picture here. So from the germinal center reaction is the source of different types of diseases that are all lymphomas, so such as Burkitt's, which I'll talk about, follicular lymphoma, which is a more indolent disease, indolent B-cell lymphoma, and also the more proeminent ones, which are diffuse large B-cell lymphoma. So this is the larger group. So Burkitt's, as surely you know, so there's been, it's a very aggressive B-cell lymphoma, so highly proliferative. So it has been firstly described by Dennis Burkitt in Uganda, so when he was seeing patients there. So one characteristic of Burkitt's lymphoma is this MYC translocation, so virtually 100% of the patients present a translocation of the MYC gene, so into the B-cell receptor gene, so that I talked about. So basically this leads to an overexpression of MYC in virtually all the cells of the lymphoma. And MYC has properties of, is a key cell cycle gene, so it makes cells proliferate very heavily, so enter cell cycle. But it has, of course, these advantages in itself because it leads to high genomic instability of the cells and a very strong cell death. So if there's no signals protecting the cells from cell death. So many years ago, we were wondering what could be the survival cue that together with MYC forms this lymphoma, so this Burkitt's lymphoma. And what we have analyzed was gene expression profile of Burkitt lymphomas compared to other non-Burkitt's lymphomas, and we found that actually there's a MYC high signature as expected, but also a PI3 kinase high signature. And we actually look into patient samples. These are not of endemic disease, but in the endemic disease, the expectation is that it's similar, that you can see, of course, MYC being very highly expressed in these samples, but then also the samples have phospho-AKT and phospho-S6 kinase, which is downstream of the PI3 kinase pathway. So PI3 kinase is a very strong survival signaling, it's a very strong survival pathway. So, and we consider that at the time that maybe the combination of these two factors is actually the fundamental cues of Burkitt lymphoma development. So we generated mouse models for this. So basically we can lead to the expression of MYC and the expression of PI3 kinase, which would be upstream of phospho-AKT, of AKT and S6 kinase. And we induce this by Cre-LoxP recombination. So these mice will only express at the stage of development where we actually induce the Cre activity. And we have generated along the years, my lab, other labs, my previous postdoc lab, generated many Cre lines that can target the development or these mutations specifically at certain genes. So, and in this case, we use a Cre line that is a SIGA-1 Cre line, which will target at the entry of the germinal center reaction. So what this means is that when these mice are born, they are born normally, but when they form germinal center reactions, they will now overexpress MYC or PI3 kinase. And what we found was that with combination of these two oncogenes led to a very strong reduction of of mouse survival, so leading to the development of a disease that very much looks like Burkitt's lymphoma. And this can be seen by the Ki-67 index, so highly proliferative, very aggressive disease. Also seen by this characteristic stereosky pattern, which is basically among these highly proliferative cells as macrophages, which actually are gobbling in the still dying cells. And you can see that in the mouse we could mimic these features that are also seen in the human. So, additionally, what we also analyzed was when we did gene expression profile of these lymphomas, what we realized is that they actually look very much similar to normal germinal center B cells, so the cells of origin of this disease. And in addition, what these mice showed is tertiary mutations, so meaning mutations that are in addition to the MYC and the PI3 kinase activation. We found, for example, mutations that stabilize another protein, another cell cycle gene, which is called cyclin D3. And cyclin D3 mutations were exactly on the same positioning that is a phosphorylation site that leads to cyclin D3 degradation. And you can see these mutations in the mouse, but you can also see it in the human. So we think that, we develop a mouse model system that mimics Burkitt's lymphoma in many of its aspects. Of course, it's a mouse, it's not a human, so there will be differences, but in this setting, it's quite a very strong and powerful tool. So, I didn't mention yet, so Burkitt's lymphoma actually comes into several types. So one is a sporadic case, which seems to occur worldwide. So there's an immunodeficiency associated Burkitt's lymphoma, and of course, as you all know, so there's an endemic Burkitt's lymphoma which is associated primarily in the sub-Saharan Africa, so and affects primarily children. So Burkitt's lymphoma has been associated for many, many years with also the presence of Plasmodium falciparum, and as you can see, there's a very strong overlap between, or this is a very old picture, so probably things have changed now, but there's a very strong overlap between Plasmodium and Burkitt's lymphoma occurrence. So EBV, which is Epstein-Barr virus, is very proeminent in endemic Burkitt's lymphoma. So virtually the majority of the Burkitt's lymphoma actually do have Epstein-Barr virus. And this actually is likely to contribute to the disease, but the function or the activity of Plasmodium potential infection onto the disease is much less clear. So we wondered together with Jean, so what may Plasmodium be contributed to this disease? So we know that Plasmodium, primarily through in vitro studies, that Plasmodium drives B-cell proliferation. This infection can induce AID expression, which is a gene expressed in germinal center reactions, which lead to also more mutations occurring in the DNA. And potentially one other aspect that Plasmodium may do is actually to induce this reactivation of a latent Epstein-Barr virus infection, something that I didn't mention. Epstein-Barr virus, of course, acutely infects, but eventually will be controlled by the immune system and it becomes latent. Potentially, a dysregulation of T cells, particularly CD8 cells, may lead to the reactivation of EBV, and this may lead to be involved in the pathogenesis. So we wanted to understand, so how does Plasmodium infection modify Burkitt's lymphoma genesis and its features? So we took the approach that basically here we are not asking the question whether Plasmodium induces Burkitt's lymphoma, because we are already using a mouse model that already develops Burkitt's lymphoma. We want to understand what Plasmodium imprints into the disease. So that's what we are trying to understand here, and we still do not know the answer, but I can just show you some of the preliminary data we have. So basically in this scenario, again, what we have is this activation of PI3 kinase and MYC in these cells, but now the germinal center reaction is driven by Plasmodium, in this case Plasmodium shabodi, and we use blood stage to infect these mice. So when we analyze now mice that are infected versus mice that are not infected, there is a slight difference. So the mice that are infected do seem to die, or do seem to develop lymphoma somewhat more rapidly. So there's a few less mice that develop the disease. It's a bit slower, but it's not striking, the difference. So both situations, either infected or uninfected, develop the disease to some extent as expected. What we notice is that in these cases, the spleens of the mice, is that the infected mice do have a very much larger spleen weight. So there's also the length of the spleens are larger, and the cellularity of the spleens, as you can see, is larger. So it's more cells, more mutant cells. And interestingly also, the cells, the lymphoma cells, do carry, most of the cells carry this germinal center B cell phenotype. So suggesting that Plasmodium is doing something and probably potentially imprinting some features on the disease. So we also notice that when we infect these mice that have this predisposition to develop the disease, which have activation of PI3 kinase and MYC, the parasitemia of these mice takes longer to disappear as compared to mice that do not have any of these mutations. And you can still observe parasites very long term. So there's still some parasites ongoing in these mice at the time that they should have been cleared out. So potentially, this interaction may lead to this exacerbation of the disease that we are seeing. So, so we think Plasmodium infections increase Burkitt's lymphoma tumor burden, so, and carrying this PI3 kinase MYC activation, so this model system. The lymphomas in mice infected with Plasmodium display an increased features of this germinal center reaction, and the parasitemia seems to remain high, so in these PI3 kinase MYC mice, and still be detected after a very long time, suggesting that maybe there's an interaction. So with the fact that the B cells are mutant and not being able potentially to produce the right antibodies to clear out the parasite. And currently what we are doing is, of course, characterizing these lymphomas, both infected and non-infected, in very high detail by exome sequencing, RNA sequencing, also characterizing the microenvironment. And of course, we want to understand now whether the features that we see, that we now recognize by looking into these tumors, can also be observed in the human, to try to understand whether there is actually endemic Burkitt's that have an imprinted signature of Plasmodium infection. So I'd just like to thank my lab. Abdu is here, so as we just mentioned before. So it's a real pleasure to see Abdu in this new setting, in this new house. I would like to thank a lot of funding from different sources, including Cancer Research UK. And I would also like to thank Nazifka and Lucy, which have also participated in this work, which I haven't shown data from them. And of course, the facilities at the Crick Institute. So thank you very much for listening.
Thank you very much, Dennis Calado, for the interesting presentation. I believe we have one or two questions for you. Yes, the first hand is already up. Second hand, okay, so Abdul Rahman and then Lasved.
Thank you, Denise, for this great talk. So Plasmodium infection is worsening the phenotype of the mouse already having somehow Burkitt lymphoma, right?
That's true.
So how important is Plasmodium infection? I mean, have you tried other infection system like to see if it will worsen in a similar way?
Yeah, I think it's a great question. So, of course, we went to study Plasmodium because that's the disease that is mostly associated, but you're right. I mean, so maybe if there are also other endemic infections in the same areas, why not, right? I mean, why would not other infections actually play a similar role in increasing the burden of the disease or increasing the characteristics that we still don't understand exactly what they are?
Lars.
Well, I have a question along the same line,
Sure.
actually, because it's not only malaria that is interesting here, but it's actually in the human, at least in endemic Burkitt lymphoma.
Mm-hmm.
It is not any malaria parasite that will do this, but only falciparum.
That's right.
So I think if we are to investigate the role of malaria in the pathogenesis of EBL, then we have to understand what is it
Sure.
that falciparum parasites can do that the other species of malaria
That's right.
parasites, and I would assume including Plasmodium cerevisiae
Sure.
in the mouse,
Sure.
cannot do
Yep.
in the role of the pathogenesis.
Sure.
I think it's
Sure.
a critical question.
Yep. No, I agree completely. I think that I would divert the question to the malaria expert. I mean, so which is in the
I would
audience.
love to discuss this more
Yes,
over a
absolutely.
beer.
Yeah.
Okay.
Yeah, thank you. So my question was sort of similar, actually. Hi, Denise. It's
Hi,
Rachel.
how are you?
So with your model, you've recreated a scenario where these lymphoma-prone mice are given Plasmodium. But have you thought about how you might approach the question from the other way round? So if you have chronically Plasmodium infected mice and then they develop mutations, would you see something similar, do you think?
So doing the infection first and then introducing the mutations
I mean,
later.
I know experimentally it's a challenge in your model.
Oh no, I think it is possible, right? I mean, so it is possible to do those kind of experiments here. So here, I think they may address actually maybe a similar question. So here we have cells that have the mutation, and we introduce Plasmodium, right? I mean, in that scenario. So the question being whether, if there is already a precancerous situation, right? I mean, would Plasmodium make it worse? Okay, so that's basically what we are addressing here.
I suppose so, thinking that malaria infection is going to be more common than lymphoma, right, in the population.
That's right,
so
yes.
Probably in the population more likely to occur that way around.
So I think the idea that, of course, Plasmodium is driving the disease, meaning it's the initiation event of the disease, is unclear. So I'm not saying whether it is or not. Potentially it's not. Potentially it's driving a precancerous scenario that is already there and creating, driving disease. And that probably would explain actually the high incidence of Plasmodium, the high incidence of BVV, but in fact only very few people will get the disease. So.
Okay. Thank you very much, Dennis. I'm sure the conversation will continue after here, where cancer and malaria meet. Thank you.
Yeah, so we will continue with the fellow session, each of which will be 12 minutes, and the rest of the 3 minutes will be for questions. We will have Dorito Bobi first, and then Kunmi after that. She'll be talking to us about the impact of ivermectin on multiple insecticide-resistant Anopheles gambisensis lato from southern Ghana.
Hello, everyone. My name is Dorothy Ubobi, and I will be presenting on a study that was conducted in southern Ghana, specifically Cape Coast, to assess the impact of ivermectin on multiple insecticide-resistant Anopheles gambiae mosquitoes. So this is just the outline of the presentation. So malaria is endemic in many countries worldwide, but over 90% of malaria cases occur in sub-Saharan Africa. Over the past 2 decades, malaria vector control has relied heavily on insecticides that are delivered through insecticide-treated bed nets and indoor residual spraying. These 2 interventions have contributed significantly in reducing the malaria burden. However, the threat of insecticide resistance is trying to reverse the gains that have been made so far. So there are 2 major causes of insecticide resistance in Anopheles mosquitoes, which are the vectors of malaria. We have the target site mutation and the metabolic resistance. So with the target site, we have 2 that have been documented in malaria vectors: the knockdown resistance, short-named KDR, and then acetylcholinesterase, which is the ACE1. And for the metabolic detoxification, enzymes that are involved in that belong to the families of cytochrome P450s, glutathione S-transferases, and esterases. Now, KDR confers resistant to pyrethroids, and then cross-resistant to DDT. And with the AC1, it confers cross-resistance to carbamate and organophosphates. Now, what is the status of insecticide resistance now? So this data covers the resistance emergence from the year 2000 to now, that's 2024. So the first graph on my left is showing the spread of pyrethroid and DDT resistance. And then the red circles and then the yellow ones are confirmed cases of resistance in those areas. So we can see that resistance of pyrethroid and DDT is widespread. And then on the right, we can also see that the carbamate and OP resistance is also increasing in those countries or areas. And what is also worrisome is that there are reports of malaria vectors that have developed resistance to multiple of these insecticides. Now, what is the impact of insecticide resistance on malaria vector control? This is very controversial because there's no evidence from a randomized control trial. But from small-scale trials, we have seen reduced efficacy of the pyrethroid ITNs and then IRS in areas with pyrethroid-resistant malaria vectors. And also, modeling studies have predicted that if nothing is done, what we have achieved so far in malaria vector control could be reversed. And so there's a need for additional interventions to complement the ITNs and the IRS to combat insecticide-resistant malaria vectors. So the rationale for the study, ivermectin is an antifilarial drug that is used by both humans and animals. And if mosquitoes ingest blood that have been treated with ivermectin, it leads to reduced survival of the mosquitoes, reduced fecundity, and even reduce a parasite and viral load. And other studies that have also administered ivermectin directly to mosquitoes have also demonstrated its impact, lethal impact on the survival of mosquitoes. However, there is lack of information on the impact of ivermectin on these mosquitoes that are showing resistance to multiple insecticides that are used to impregnate bed net and for IRS activities. And so the aims of the study was to investigate the impact of ivermectin on the survival of multiple insecticide-resistant Anopheles gambis and Sulato population in southern Ghana. And the objectives were to perform mosquito sampling and bioassays, determine the composition of the mosquitoes that will be sampled, and also detect or genotype the KDR mutation and then the AC1 mutation, as well as to estimate the enzymes associated with metabolic resistance. So we collected mosquito larvae, that's Anopheles larvae, from 4 areas in Cape Coast. And then we transported them to the University of Cape Coast, where they were reared to the adult stage, and we used them to perform ivermectin and WHO susceptibility tests. The mosquitoes from these tests were transported to Noguchi for molecular analysis, and then we also sampled some for biochemical assays. And the study was, the study period was between May 2023 and then June 2024. So for the laboratory assays, for the bioassays, what we did was to set or decide on 5 concentrations based on previous studies. And what we did was to feed the mosquitoes in all, a total of around 1,400 mosquitoes were used. So we fed them with ivermectin that have been dissolved in 10% sugar solution, which is usually used to feed the mosquitoes in the lab. And then we added a dye as a marker, just to identify those that fed on it. So what we did was to starve them for 12 hours, then we fed them for 12 hours in paper cups. And those that fed were transferred to new cups, and they were observed for mortality every 12 hours for 48 hours. Then we went on to perform WHO susceptibility tests. We exposed them to the discriminating concentrations of 5 insecticides from 5 of the insecticide classes, according to WHO protocol. So we observe them for an hour after exposing them to the insecticide for an hour for knockdown, and then for mortality after 24 hours.
You have 5 minutes more.
Oh, okay, so we went on to also genotype them to identify the mosquito species, Anopheles species, and also to detect the resistant mechanisms involved. Then we also performed the biochemical assays to estimate the enzyme activity. So for the ivermectin susceptibility bioassays, this is a box plot showing the percentage mortality recorded at the different time points after feeding them with the ivermectin. So looking at the plots, we realized that as the concentration increased, the proportion of dead mosquitoes also increased. And it's the same way as the concentration increased, the time needed for the mosquitoes to die also reduced, indicating that ivermectin is dependent on concentration, higher concentration, and then time also, the duration of time they are exposed to. I'll skip this one. And we also performed the susceptibility test. So this is the result of the susceptibility test. The population was resistant to the pyrethroids, that's permethrin and deltamethrin, as well as the DDT, that's the organochlorine. However, they were moderately resistant to the bendiocarb, which is the carbamate, and then the malathion, they were susceptible to it. So at the end, we genotyped 220 of the mosquitoes from the test, and 201 were confirmed as Anopheles gambiae, and 58.2% were found to be Anopheles coluzi, and then 27.9% to be Anopheles gambiae SS, showing that the steady site is dominated by Anopheles coluzi. So for a KDR genotype, the overall frequency was reasonably high, around 71.79%. And for all the other groups too that we calculated the frequency for, we realized that the frequency was higher in the dead mosquitoes than the alive one because it killed more of the mosquitoes. So we weren't getting more of the alive ones, showing that it's more effective against mosquitoes, even showing resistance. I think, okay, sorry. I don't know why. I really don't know.
Are you trying to get back?
Yeah.
Technical team. Okay.
Yeah.
Okay.
I skipped one.
I think you have to slow down from here.
Okay. Yeah. So
We have
with
about
the
1 minute more. So
okay,
yeah.
so with the AC1, the overall frequency was low, and this correlated with what we saw in the phenotypic assay, that the pyrethroids were resistant, and the population was resistant to the pyrethroid, but moderately resistant to the carbamate and then the organophosphate. And with the biochemical assays, comparing the test mosquitoes or the test population to the reference strain, which is the susceptible strain, realized that they showed high enzyme activity for all the enzymes that were tested. Yeah, so to conclude, these few findings, I want to go through these few findings. So from the study, we realized Anopheles gambiae from the site was highly resistant to insecticides from the 4 major insecticide classes. And then the data demonstrated ivermectin's potency to control mosquitoes with multiple insecticide-resistant mechanism. And we recommend that additional studies should be conducted to ascertain the appropriate dose, administration technique, and the impact of ivermectin on other mosquito population. Thank you.
Thank you, Dorothy.
Any questions? I have, but if we can have from the floor first. Okay, just I have about 2 or 3. First, you said Southern Ghana.
Yes.
And Southern Ghana cuts across 4 regions. Meanwhile, you were working in Central region. So why Southern Ghana?
Okay.
I mean, that's just
yes.
for you to think about.
Yeah, okay.
Then we're talking about ivermectin. You know the mechanism of action of ivermectin is different from that of the other insecticides, which was both the organophosphates and the pyrethroids, together with DDT. So, were you expecting that you have a result that is different at all?
Not really, but I was just trying to test to see if those that are resistant, they will still be able to control. Because though it has a different mode of action, if it has not been tested, we wouldn't know.
Also, this thing is just because it hasn't been
Yes,
considered.
and then, to know if it will be able to control. There have been so many studies, but it's just exposing them to it and not telling us whether they were resistant mosquitoes or not. But I wasn't expecting them to be resistant to ivermectin because it's not even implemented yet.
Oh, I understand the last speaker isn't available.
It's online.
Is it online or not available? Who says it's not? Oh, okay. Now the other, sorry, I'm just ignorant since we are closing after this. Yeah, is there anybody else who has a question before? Yeah, please. Can we have the mic?
The mode of administration of testing ivermectin, in this case, you actually fed them with ivermectin. In terms of mosquito vector control, I think the modes of application do not really require us to feed mosquitoes. The insecticide or the anti-vector agent is applied on bed nets or on walls. So what do you think will be the best ways of, because you've shown that they're effective in killing, but this is through ingestion. So how do you expect to, what it would be a suggestion moving from what you've determined now experimentally to actual translation and testing against mosquitoes in the natural world? That's my first question. The second is, you said the species that you had were mostly Culizae. Did you determine these using molecular methods or was just morphologically? We are finding out that the mosquito species around are more complex than we see with the eye. If you go to the genetics, there are other subspecies or some cryptic species that are emerging now. We don't know whether these same mechanisms are operating in them or whether they would also be susceptible to ivermectin. So I'm just asking which method to use for speciation.
Okay, I'll start with the last question. Yeah, so we use molecular analysis here to identify the Anopheles coluzi. We didn't use morphological identification. And for how the ivermectin will be deployed, I think my work is actually on attractive toxic sugar bait. So this is an aspect of it that I'm trying to test the efficacy of the toxin. So the idea is to deploy it in attractive toxic sugar bait. Since now we don't really have a clear outdoor intervention to control mosquitoes, and then using ivermectin and deploying them, sometimes it is sprayed on vegetation around homes, and sometimes it is also put in like a container or something, set outside. And even now, they have moved to the commercial level where they are also manufacturing other ways and other means to deploy the attractive toxic sugar bait. So the idea is to test the efficacy of the ivermectin, and if it's good, and then use it for attractive toxic sugar bait, if it's effective. Yeah.
So you mean farmers are using this?
No, it's under trial now. Yeah.
Okay, well, thank you very much. Oh, was there any other hand? I'm sorry. Okay. Thank you all very much for your cooperation.
Thank you, Reverend and Anastasia. So, we finished ahead of time. Who would have known? All right. I think it's been a day of ups and downs, but we finished well. And as is the tradition, there's the option of loggia after this event. Those who know, know. So loggia is basically faculty, for want of a better word, drinking spot, watering hole. Faculty watering hole, and typically after, especially the first day of the conference, faculty and invited guests converge there, and we talk about intellectual and non-intellectual topics. Yes, rubbish. So, because the weather is a little bit, we have South Africans who brought winter with them and the British people. So the weather is a bit gray outside. So we were wondering whether people were actually interested in going to loggia today. So maybe by show of hands, and if we don't have enough hands, then we'll postpone loggia to tomorrow. So show of hands, those who are interested in going to loggia. Not the student fellows, faculty and invited guests. All right, so I think we'll do loggia. And today was, as I mentioned, dedicated to mostly host immune responses and disease pathogenesis, host pathogen responses, which will also happen on the last day. Tomorrow will be a day we'll have molecular epidemiology, also drug resistance, drug discovery mechanisms and resistance. And then for every subsequent day after today, we'll have 2 keynote speakers for the day. And tomorrow the keynote speaker is going to be Samuel Mauli Adade, who is an alumnus, and you heard from him today. And then also Professor Basiru Bonfo from Afrique 1. So I think I'm keeping us from our water. So good evening and see you all tomorrow. We're done for today. And one last thing. Tomorrow we start at 9 o'clock sharp. And we will be here. We'll start every day is now here. we have this room, and then there's 2 overflow rooms, which is the boardroom and the PhD lecture room. So if you come and this is full here, we have those overflow rooms.